Good morning, everybody. Good morning. It's great to see so many old faces as well as new faces here. People come for the first time. Welcome to the 24th Annual BC Marine Mammal Symposium. I'll talk in just a minute a little bit about the background, but first as I think many of you already know, last night Aurora died. And we'll talk a little bit about that later this morning. Lance will tell you what we do know and answer questions as well. And so that's going to happen in the agenda under the section on education, museums, and aquariums. We've had a few cancellations, unfortunately, this morning, and the first two under the aquarium section have canceled. So Lance will be speaking at that time. The Marine Mammal Symposium. We've got a lot of people here in the room. In fact, when we started this, it started with an idea. Jim Darling and I were attending the Marine Mammal Conference, the big international one. And it was 25 years ago, and it was in Chicago in December. We happened to be on a yellow school bus being driven to the Shed Aquarium. And we had a crazy bus driver. To get around traffic, he was driving down the sidewalks. And there was back-to-back traffic, and it gave us a lot of time to talk. And one of the things we spoke about was the fact that a number of us were doing research in marine mammals, and we were sort of seeing each other only once a year, or once every 2 years, in fact, because we were going to this big international conference that drew everybody. We thought, well, why don't we do something on a smaller scale that's just for us? And the us, of course, has been researchers. But the us is also the whale watching fleets. The us is the Vancouver Aquarium. The us is facilities that help care for marine mammals. The us is really big. And we decided we wanted to do something that was not the traditional scientific conference, but was one where people had a chance to get an update, to hear sort of brief snippets, not the long, detailed scientific talks which we get at the more formal conferences, but something more in the highlights. If somebody really wants all the fine details of how you look for sea lion scats and clean it, you can talk about that over coffee. We just want to focus on, so what do we find? What are we doing? What's neat and new that we didn't know about before? So we started this, before this building was here, there were Second World War army huts, some trailers, and we started with about 30 people that came the first time. And today we've got over 230 people that are here. And so it's gotten bigger and bigger each year. Last year, when we were leading this, we recognized that we now surpassed the size of this room. And we had two options. One was to leave this building and go into one of the formal lecture theaters where really kind of like first year of university, you're all alone, nobody knows you. What am I doing here? And so we thought a better option was to stream this into the classroom. So we have an overflow, recognizing that some people have to come, they can't stay the whole time, and it gave us that flexibility. Marcus, who was down here running the show for us, kindly volunteered to come in and said, oh, no problem, I can make this work. In the process of streaming it, we were using YouTube Live. And while we had announced it to the world, people were tuning in, and they were picking it up first by accident and then spreading it very quickly. And as Mark just says, we currently have 40 people watching online as well. And so I realized that, well, not everybody can make it each year, and there is interest beyond these walls as well in terms of marine mammals. So we decided to do something which is quite different, which is to stream our symposium so that those who want to see it can watch. Science has changed a lot in the 25 years that we've been working to make this symposium happen. It used to be that scientists only spoke to other scientists and what we knew stayed within the room. But as we're now discovering, science is being tweeted in real time. Images that people show in conferences are being posted by members of the audience, and members of the press are now attending and reporting on what's being said. And so along those lines, we've got Larry Pinn from the Vancouver Sun, and who finds this a great way to hear what's going on and to speak to people about it. So it does mean in the end that we are thinking through how we present our science, knowing that the whole world is listening, and nothing is off the record. And so just for those who are speaking, keep in mind that we have a huge audience now, and which speaks well to the science we're doing, the observations people are making, and the activities you're involved in. People want to know about it, and this is a chance to share that. So the agenda then is set up such that we have a lot of time for breaks so people can talk. That's the other value of these meetings, is meeting people, perhaps meeting people you don't know, meeting some of the speakers. I would encourage the speakers after the sessions to come down to the front. Nothing worse for me than trying to identify a speaker afterwards. I can't find them, and I really would like to have spoken to them. So please, speakers, remember to come back down so that you can continue some of those discussions. Two more minor points before we start. So you can bring water in the room, but no coffee or juice drinks, please. No food in the room. The food should stay outside. During the breaks, you're welcome to go to the Beatty Museum to see Big Blue. And there's no charge for you to go in. Please let the people at the desk know your name tag will be your admission to go in. And there should be ample opportunity, primarily at lunch, I think, or at the end of the symposium to go over. I think it's well worth it. I think that's all the formal announcements I've got. I should have said my name is Andrew Treit in the beginning. I'm a professor here at UBC in the Institute for the Oceans and Fisheries and director of the Marine Mammal Research Unit. So with that, I'd like to call up our first speaker, Carla Crossman, UBC alumnus, and did her master's here and is continuing her line of research.
I know this has directions on it. Okay. Oh, perfect. So, hi everyone. I'm going to talk today about some of the work that myself, Alison, Miss Campbell, and Lance Barrett-Leonard have been working on in the Vancouver Aquarium's Conservation Genetics Lab, looking at adaptive variation in killer whales of not just BC, but also Alaska. So in total, we're actually looking at 12 different populations, and these populations have all been previously identified by some of the work that Lance has done and some of the work that Phil Morin has done as well. And so we're looking at 4 populations of residents, 7 different populations of bigs, and a population of offshores. And between these populations, we're interested in comparing levels of adaptive variation. And when I say adaptive variation, I'm talking about genetic variation in an individual that confers some kind of selective advantage. So a situation where it would be beneficial for an individual to have lots of variation in their DNA. Yes, it's 9:30 a.m. Yes, we're talking about genetics. It's not going to be scary. But now I'm going to say major histocompatibility complex is the area we're looking at. MHC is the region. It's just an important region in the DNA that codes for a lot of immune function. So it's really important that there's a lot of variation at this site. In particular, we're looking at a specific region called the DQB that codes for extracellular protein receptors. But I'm going to explain why it's so important to have this variation. So genetics 101, you might have a blue whale, you might have a red whale. They can mate and produce, you get half your DNA from your mom and half the DNA from your dad, so you might get a whale that's both red and blue. And now we're going to go through an example of 2 populations, one on the left, one on the right, of whales. The first, you might get some red whales, you might have some blue whales. The second one, we've got red whales and blue whales, and some whales that are both red and blue. Now, imagine we had a virus, a pathogen, a disease of some kind come into the population that red whales were really susceptible to, and they die. Viruses are happy, but you can see that it really changes the look. And imagine the same thing were to happen in a blue disease or something that was blue whales were susceptible to. You can really see how it's really beneficial to have a lot of variation for yourself. But there's also benefits to the population as a whole, where increased variation enhances population viability. So with lots of individuals that have that variation, the population can do a little bit better. And so when we're looking at this, we use DNA, so you get 1 copy from your mom, 1 copy from your dad, and when we want to quantify these levels of variation, we're looking at what we call variable sites. So areas where what you inherit from your mother is different from what you inherit from your father. And when we average these out between all the individuals in the population and compare this across all 12 populations, we get to see a little bit of the patterns in these comparisons. Now, this is really important. We're interested in the relative levels of variation. We're not necessarily interested in absolute values. And don't worry, there's lots of differences here. It's nothing to panic about. Yes, some small populations like AT1, the bigs, and the southern residents have very little variation. In fact, we didn't find any variation in the southern residents at the region we looked at. But a lot of other populations had quite high levels of diversity. So, but this small populations and low levels of diversity is actually something you kind of expect. In small populations, inherently, you do expect low variation. And what we found with southern residents having little variation is consistent with, it's kind of a third piece of evidence, consistent with other work that Lance has done and Phil Moore, and Mike Ford has done, Phil or Mike, I'm confused. One of them have done, pardon? Mike, that Mike Ford has done, looking at showing that there's very little genetic variation in southern residents. But this, again, is only one region of the DNA and only one region that controls for adaptive variation in the results that I presented here. So we're interested in looking again at adaptive variation at a different site to see if the patterns are consistent or to see if maybe there's something different going on. And so next time we're going to look at intracellular protein receptors, so just a different way that the immune system might work. So overall, we found different populations having differing levels of variation and identified some populations with very little genetic variation. And this is kind of somewhat consistent with some of the other studies, but we're really interested to kind of move forwards, expanding what we've looked at, not just at this one region, but looking at more and different metrics of variation as well. So I need to thank the Wild Killer Whale Adoption Program at the Vancouver Aquarium. If you're looking for that special gift for that special someone, you can always adopt them a killer whale. Generous donations from Mr. Doug Horswell, who made all this work possible. Thank everybody.
So we do have time for questions. If anybody would like to ask Carla a hard genetics question? Maybe I can just ask you, so which populations have the most genetic variation?
So it's going to depend on what metrics of variation you look at. So I just talked about one. I screwed up. No. That's okay. I think some of the Eastern Aleutian transients or Eastern Aleutian residents had some of the highest levels of variation we saw using this metric and using a couple other metrics that I didn't talk about. So there we go. So yeah, Eastern Aleutian bigs and offshores are also, Southern Alaskan residents had kind of the highest levels of variation using this metric, but there are other metrics that we're using that kind of change these patterns up a little bit.
We're going to pass the mic, and I think I've got a runner here for the mic.
Now you're talking about variation at one place in their genetic information.
Sure, yeah.
And it's in relation to their immune system. Do you know, is it a specific aspect of their immune system or their entire immune system?
So the specific region we're looking at codes for a subset of extracellular protein receptors. So it's one small aspect, a tiny aspect of how it works, and it's only a subset of these extracellular receptors. So there's lots of different ways our immune systems are incredibly complex. So it's just one small aspect of that adaptive variation. There could be great news in other areas, so do not panic. It's just interesting that we found this, and it is kind of consistent with other lines of evidence.
Carla, can you use ancient DNA to compare extant populations with past populations?
I'm sure we could, if the samples existed, and I'm sure they do somewhere. And that's kind of one thing to note here is that these samples weren't all collected at one point in time. So some of these samples date back to, I believe, late 80s, early 90s, and onwards. So, putting this in a time perspective, these are long-lived animals, but it would be interesting to be able to use ancient DNA to look at, changes over time if we had access to those samples, for sure.
And our next speaker is Joan Lopez.
How will I forward? Okay. All right, I'm not sure why Andrew put me into the science section. This is mostly, well, there's a little bit of science in it. Basically, what we're seeing is a lot of changing trends in our whale encounters in the Salish Sea. And that's sort of a rundown of what we saw this year. Now, when I say 101 days with transient or big killer whale encounters, we might have seen the same group of whales twice in the same day. We count that as one. There may also have been overlaps there where we saw transient killer whales and southern resident killer whales on the same day. Those would, of course, be counted as different things. But this is the first time, I've been doing this since 2003, it is the first time where our transient killer whale encounters far exceeded those of the southern residents. Now, that might have been partly that the southern residents weren't around that much this year. I'm sure we're going to hear more about them as the day goes on. But the big story is the transient killer whales were there on such a frequent basis today. Our humpbacks are down a little bit, but Victoria will tell you more about humpbacks, I'm sure. Also of note, at least 30 more, actually, 30 plus encounters with the transient killer whales involved in excess of 10 animals. So this is also a big change. They're not just skulking around in little groups anymore. Sometimes there's a lot of them out there. Okay, this is the only sort of, I'm going to bore you with a couple of tables, but what I'm trying to show you here is a little bit of a trend that we saw. First of all, the way transient killer whales are numbered, they all start with the letter T. Initially, they were all individually numbered. They got up to about 200 animals, realized that was not going to work. Thank you, early whale scientists, for coming up with the alphanumeric numbering system, which is much, much more convenient. But there is still those old numbers linger. For example, the first three that you see up there, the females are sisters. So if you don't go and look at the old history of the family tree, you'd never tease that out. The T36A, T99, and T137 are all sisters, but they are. I've also indicated on the tables how many animals are in the family group. If it's a loner or two non-family traveling together, I've indicated the genders, or if it's a juvenile. So what I was trying to show with this table, because I think there's a trend, and maybe there's some aspiring master's student in the audience who'd like to tease this out. There are some family groups or individuals that seem to have fairly good site fidelity to the Salish Sea area. So this is mostly Georgia Strait and Gulf Islands, but we did extend down as far as maybe Constance Bank, maybe over as far as Anacortes on these numbers. There are some animals, for example, the T123s, which show up most frequently in the spring. And as we go on through, you'll see there are some individuals that are rare in the area, so they must have a good site fidelity maybe somewhere else, and some animals that have more site fidelity as we get into the fall. So this is the next group. As we're coming along here, right at the very bottom there, T51 and 49A2 is one of those unusual groupings, a male traveling with a juvenile, and T51 is not common to our area at all. And then as we go along, now we're getting into the animals that show up typically more late summer and into the fall. Now, this is one company. It's our data. If you go and you talk to another whale watching company, their data might be slightly different. And if you go and you talk to the people from Victoria, again, their data will be slightly different because they are traveling in different areas, seeing whales maybe at different times. But there seems to be a little bit of a trend there. Now, it's not just this year. I can look back at my data for the past 2 seasons as well, and I'm seeing this repeated trend of certain animals showing up at certain times. Just another one of those funny family situations. T34 is the mother of T37, who of course is the mother of 37A and B. If we get those animals together, we have 4 generations, and that did happen this year, 4 generations of transient killer whales traveling together. So although they're very fluid in their dynamics and who travels with who, quite honestly, there is a certain amount of family fidelity there as well, even across generations. Okay, enough on the numbers. The rest is pretty pictures. That's T51, and the way we recognize killer whales, of course, many of you know this, is by the dorsal fin and the saddle patch, looking for unique marks. He has a nice little nick right at the top of the fin and some really unique scratches. 2003. I've never seen him before. So he's unusual in our area. This is another one of those weird pair-ups. T2B, who is, by the way, an adoptable whale, Petter, is an older female in her 30s. She's never had a calf. She usually travels with the T60 group. Well, this summer, she was off on her own with T60D, reported to be a male, about 9 years old. I don't know, we had a lot of cougar jokes flying around this summer about that. And they weren't in some place where it would be really easy to find the rest of the T60s. They were in Saanich Inlet, they were in Samson Narrows, they were in these out-of-the-way little places that they probably weren't going to encounter the rest of the T60 family, who were out and about at the same time. So it was just one of those little interesting things. Did they get lost? Did they do that deliberately? I don't know. We can't get into their minds. Just introducing you to a couple of newbies this year. T86A4 was probably born through the winter. This picture was taken in May, and you can see nice, healthy plump and no orange. T100B2, that little cutie. This picture, the little guy might have, or gal, might have been about 1 week old when the photograph was taken. Interesting thing, T100B and her older offspring had separated from her mother, from T100. She was off on her own. All of a sudden, she shows up again with mom. Oh, that's cool. And about a week later, new baby. She stayed with mom for about a month, and then off she went again and dispersed. So I don't want to anthropomorphize here at all, but we can kind of make a little bit of a comparison of that family care when a new offspring is on the way. And of course, what transients? What's the big deal about transients versus southern residents, or any residents for that matter? It is the diet. And face it, we need these guys in the area. Our seal population is large, but it's not growing, thanks to these guys. These females can have a pup every year, the female harbor seals, but the population's staying about the same. So sometimes it's easy. This whale, well, this whole group of whales actually just had to wait for the tide to come up, which it was doing, and those seals had to swim. Why work hard if you don't have to? But sometimes you do, right? This was another harbor seal hunt, and actually in that case the harbor seal survived, but that's a story for another day. But yeah, sometimes they have to work hard at it. Harbor porpoises, I'm afraid, did come under fire a bit this year. I know it's not a very clear picture, but if you look carefully at the front of the killer whale, you can see a little harbor porpoise that was possibly taking its last breath, or maybe was already done at that time. So that would explain what the bouncing around was that we could see from some distance away. There was harbor porpoise pursuit. I wish I had gotten the pictures from my colleague of Adol's porpoise pursuit. They are on our Facebook page, a little kick out there for Vancouver Whale Watch. Whales were airborne, absolutely airborne. So yeah, it was that was pretty amazing. And of course, sorry you sea lion researchers, but sea lions are also on the menu. Now we know this is a sea lion because the whales actually pushed it over to the boat to show us what they had. Yes, thank you very much. Passengers were thrilled. And you could still see the flippers. They still actually had skin on them. Nothing else did. And the gulls were having a great time. But after a meal, there's usually time for socialization and play. They do interact. And speaking of play, turns out they like surfing those standing waves in passes. This was an active pass, T65A group. And there they are again. Actually, there's a whole series of pictures. The whales don't move. We also saw them do this in Dodds Narrows, which also has a really good tidal run and good standing waves, and at Boundary Pass. Now, there's no reason why those whales can't exit that wave. They chose not to. They just stayed in that standing wave until they were tired of it, and then they left. So good for them, having fun. Surfing waves in Georgia Strait as well. There's some more porpoising whales. That was the day of the Burns Bog fire, incidentally, if anybody cares. But really, this is kind of why we do these things, these beautiful moments. That just nothing beats that scene for our passengers and for us of these amazing animals. Thank you to my colleagues for identifying and faithfully recording all the whale sightings so we can kind of put together these little patterns. And also thank you to the whale research people at DFO and at Center for Whale Research for continually updating those guides, the identification guides, so that we can continue to get to know these animals, recognize these family groups, and hopefully contribute a little bit to the science in the future. Thanks. And there you go. Wow, I didn't even push the wrong button. Questions?
Oh.
Said, is this on? So you said with the whales, like the 51, you don't see very often. You said they might have areas that they're sighted more often. Do you ever collaborate with other kind of whale watching that would have sightings? Kind of track where the whales are going?
Yeah, that's a good question. We do a bit amongst the area. Certainly, we have our own sightings network that's quite informal. But it might be a whale that is more likely to be seen, say, in the Campbell River area, Tofino, Prince Rupert. Your base for all of that, and I hope everybody reports here, is the Cetacean Sightings Network. That is where all the data is. And where, like I say, my company, it's my data, it's quite unscientific at this point, but if anybody were interested, that's the source, and they're collecting it from all over the province, and I'm sure whales are being identified, thanks to all those beautiful guides, all over the province. So I think the information's there.
Okay, and then we have one question that's coming from Amber online. She's watching on YouTube, and she is just asking, did you measure what behaviors the groups of killer whales were engaging in? For example, were the larger groups engaging in hunting or social behaviors, or tend to contain calves?
Oh, okay. The larger groups did engage in hunting behaviors, which, you know, they usually seem to be sort of the stealthy sneak up on their prey, but not always. Sometimes an animal's just in the wrong place at the wrong time. So yes, hunting behaviors were observed with big groups, but often it was very, very social as well with the larger groups.
Maybe I'll ask one, which is, do you see any effect of the vessels on the transient behavior? Are the vessels aiding in the hunting procedure by pushing seals off, or is there no effect? What do you think?
Okay. We try our best to not interfere in any way. So if the whales are hunting, the vessels actually try to back off, and also most of the, just to put a little caveat in there for things, most of the whale watching vessels in the Vancouver area are jet-driven boats, which are at low speed, extremely quiet underwater. And that's probably the biggest thing for the transient killer whales. They hunt by listening to the sounds their prey makes. It's one of the ways they hunt. And that very subtle surfacing of a harbor porpoise or the splashing of the seal, they have to be able to hear that. And we do see them hunting, even while we're watching them. At this point, I don't feel that we are either helping or a deterrent either way.
Thank you.
Welcome.
Thank you. Our next speaker is Selina Agbayani, who is a new master's student here at UBC.
Good morning, everyone. Thanks for coming. My name is Selina Agbayani. I am a first-year master's student here at MR. I started in September, so everything is very new, and I'm here to share with you what project I will be working on. And so before coming to MMR, I had a background in terrestrial ecology. I have a bachelor's in forest sciences from UBC, and I went to IT to get an advanced diploma. And after BCIT, I had an opportunity to work with World Wildlife in Canada, and it was there that I was introduced to the world of whales. And here at MMR, I will be working on modeling the migration of the eastern Pacific gray whale in the face of climate change. So most of you have probably already met the gray whale, but if you haven't, this is what they generally look like. The adults get to about 11 to 14 meters in length. And what's interesting about the grays is that they're what we call benthic feeders. So they feed on benthic invertebrates such as amphipods, which are found in soft substrates, and mysids in rocky reefs. Now, they generally feed in shallow areas, and so they stay pretty close to shore. And this pretty much increases the chance of overlap with human activities. Sorry about that. Is that better? Awesome, thank you. As with the other great whales, the greys were historically hunted almost to extinction. And today there are only two populations left, the Western North Pacific and the Eastern North Pacific. In the 1970s, the greys were assessed as endangered and were afforded some legal protections. This was great for the Eastern North Pacific population. That population started to rebound, and in 1994, they were delisted. While the Eastern North Pacific population recovered pretty quickly, the Western North Pacific population didn't. If you look at the habitat on the left, that area is actually quite huge, but there aren't very many whales left using that habitat. It is interesting to note, though, that recently there were some individuals from the Western North Pacific population that were seen to cross to BC and breed down in Mexico. So the greys are known to have the longest annual migration of all the greys, and they spend the summer up in the Chukchi and Bering, and then they come back down to Baja, Mexico in the winter to the birthing lagoons. And then they come back up again in the spring. And overall, that's approximately 15 to 20,000 kilometers round trip. The northbound migration starts in mid-February, and they reach the Bering Sea around mid-April. Migration itself is pretty interesting because it occurs in pulses by sex and age cohort. The pregnant females go first, followed by the other adults of both sexes, and the juveniles of both sexes. And the mothers with calves leave last. Now, the mothers with calves are pretty interesting because they actually stay closest to shore compared to the other cohorts, possibly to avoid predators. It's generally known that most of the feeding occurs up north in the Arctic. And we don't think they feed on the southbound migration, but they have been witnessed to feed on the northbound. So they've been doing pretty well. But in 1999 and 2000, there was an unusually high mortality event, where up to 1/3 of the population were reported to have been affected. The stranded whales were emaciated and malnourished, and so this particular event was largely attributed to starvation. This phenomenon led me to some of the questions that I will be asking for my thesis. Why do they migrate in the first place? How much food do they need in order to complete their annual migration? I'm also interested in seeing whether or not climate change affects the amount of food available to them, and whether or not that will affect their ability to complete the annual migration. Over the next little while, I will be working with bioenergetic models and spatial movement models in order to address some of these questions. If you have any suggestions for me, please feel free to come and chat with me after. Thank you.
I'm not sure if anybody has any questions or, oh, there we go. We got one here.
How many populations actually do that entire migration up to the Bering Sea, or are there other ones that do partway up the central coast and then stay there for the summer and then come back down?
Well, I believe that a portion of the population has been noted to stick around. They've been called the Pacific Feeding Group. PFG, Pacific Coastal Feeding Group. And we know a little bit about them, but that's one of the things one of the portions of the population that I'll be looking at as well. So.
And you bring up a good point, because COSEWIC is in the process of reviewing the status of gray whales. And one of the things that's being discussed a lot is whether or not we've got one population or two. The second one is this coastal feeding group. They're known to be resident, repeat sightings of them. So COSEWIC is considering whether or not that should have a separate designation from the overall population that does go further north. Of course, the complicating factor are the ones that are coming from Russia across to BC and going down and now being seen to breed in Mexico. So it's been a part of the struggle of, once we put on some satellite tags, again, understand just how complicated the movements and population structures are. Then, of course, once some of those whales were seen via the satellites, they then started looking at some of the catalogs and discovered, in fact, these whales have been seen long before satellites were tracking them. So those are some of the things that COSEWIC will be dealing with, how to come up with the proper designation and the proper population designations as well. We'll move on. Thanks, Selena. Next, we have Sarah Fortune.
Just a reminder to all the presenters, please try to speak into the microphone. Remember, there are people listening.
Thanks for the reminder, Rob. And thanks for the introduction, Andrew. So I'm going to take everyone further from home up to the eastern Canadian Arctic. That's where I've been studying the foraging ecology of the eastern Canada, West Greenland bowhead whale. Now, those of you who know me know a little bit about the epic failure I experienced in 2015. This was supposed to be my last field season. I was supposed to collect this really important information about bowhead whale feeding behavior and diet. And instead of collecting any information, I spent the summer on land. That's because we had unusual amounts of ice that persisted throughout the entire summer. Here's a little aerial video of the ice. This was blocking the fjord. There's a little bit of open water, but this is the mouth of the fjord. All of these fishing boats were stuck in the fjord. Now, luckily this year, the weather gods were much kinder to us, and we were able to conduct our field work. We didn't have bad ice, and we had quite a few whales that were accessible by boat. And in order to study their diet and their feeding behavior, we equipped several whales with non-invasive short-term tags, and these record the underwater movements of the whales. And we also collected information about their physical environment and also about the prey base, but I'm not going to talk about that today. So just to give you an idea of the type of data we can collect using the fine-scale tags, this is the dive profile of one whale that was tagged for about 13 hours. And on the y-axis is the whale's depth in meters, and then on the x-axis is time. And we've got some dives that occurred during the day and others that occurred at night in the shaded gray area. And so the main thing you can see here is there's a lot of variability in terms of where the whale is diving to. And the important part for my study is to figure out which of these dives reflect feeding events. And if we zoom in on just a portion of that data, you can see that the whale conducts, relatively deep dives, going down to 300 meters. Sophisticated animations. Now, it may seem like 300 meters is pretty deep, but for a bowhead whale, this is not impressive. Many of the whales have conducted much deeper dives, down to 700 meters in some cases. Now, for these relatively deep dives, the whale spent between 15 to 30 minutes subsurface. Also, to your eye, that may seem like a long time, but it's really not that impressive for bowheads. We do have dives that have lasted 60 minutes onwards to 70 minutes. But what was a bit interesting was that following these deep dives, we found a series of shallow dives that were of, considerable duration. And we have a couple hypotheses for what could be happening here. And I think the most probable one is that the whales are locating prey at two different layers. So I believe that they're finding a deep water prey source. Probably it's high in lipid content. It's worthwhile for the whales to dive deep, but then they're also exploiting a subsurface aggregation of prey. And this is something we'll easily be able to test as soon as we get our fine-scale prey data back. Believe it or not, but it's actually still stuck in Iqaluit to Nunavut right now. It's been there for the past 3 months, and we're waiting for it to be shipped down south. But we do have information about the vertical distribution of prey in the water column. So we'll easily be able to test these hypotheses and determine where the whales are feeding in the water column. Now, while we are also in the field, we are really fortunate enough to collaborate with the Bowhead whale hunters. So this year they are harvesting 1 whale with a subsistence hunt. This is the community of Pangertung where we were based. And we spent 5 days on Kikiktuk, which is a historical whaling island. It's an uninhabited island. But this is where the American and also the Scottish whalers were based in the late 19th century. And here you can see a picture of an old Inuit home and the structure of the home that was built with some wood, remnant wood, and also some bowhead whale rib bones. And now if you walk around this island, you can find lots of artifacts from the old whaling days. So here we've got some anchors. There's also some oil processing tubs. There's actually an old above-ground burial site. So it's a very interesting location. It's a historical site, and it's where the current subsistence hunt takes place. There's also many remnants from bowhead whales and other marine mammals that have been harvested. So we were very lucky in that we were able to collect samples from the harvested whale, and this presented a really unique opportunity to collect really valuable information, to answer questions about the species that otherwise we would not be able to answer. So we collected things like skin and blubber from the whale, and this provides information about the lipid content and also can be used for stable isotopes. And that provides us with information about the diet of the animal. We also collected measurements on reproductive organs, on baleen and morphometrics, and most importantly for my work, we collected stomach contents. And this provides a fantastic opportunity to actually know what the whales are feeding on, something that otherwise we don't have the opportunity to know. And what we found with this whale was that it had consumed Arctic copepods. So these are small crustaceans. They're about the size of a grain of rice. They're really high in lipid content. And they're the same organisms that we were sampling in areas near bowheads. So this was really great because it showed us that, yes, we are sampling the food that the whales are eating. So it was definitely very helpful for our research to confirm the samples that we've collected. And so in terms of conclusions that we can draw preliminarily, the first is that bowheads appear to employ a flexible foraging strategy. So they appear to be able to forage at depth and also in shallower waters during August in Cumberland Sound, Nunavut, which is where our work was based. And they also seem to allocate a considerable amount of time to foraging activities. So we've observed what we believe to be feeding dives during the day and during the night. And we also believe that their primary prey, at least in this region, are these Arctic copepods, the calanoid copepods. Now, as with all projects, this project was very much a collaborative effort, and I'm very appreciative of all of our contributors, particularly Bill Koski from LGL Limited, Brian LaForest from World Wildlife Fund, and Thomas Seitz from VDOS Global. So thanks very much.
Any questions for Sarah? Say what? Oh, we get 2 online questions. There we go.
Okay, let's start with those online questions. The first one coming from Valentina, and she's asking, first of all, she says, great talk, and then she wants to know, how were population trends during the unusual mortality, and had they reached carrying capacity?
Oh, great question. So since the whales are almost decimated from commercial whaling, they have rebounded quite well. So it's not to say that they've reached pre-exploitation stock sizes. but their population is increasing quite a bit. And right now, I believe the population is over 6,000 individuals.
All right, and we actually have a second question, and that one's coming from Leonardo, and he's asking, is there a way to actually know that the whales are diving so deep to feed? And also, how do they choose where to dive for prey?
Yeah, also a great question. So once I have my prey data, I'll be able to map the density of food in the water column and overlay that with the depth that the whales are diving to. And that will provide a really nice visual of the prey field and will really determine which dives are foraging dives and which are not. But even without prey data, we can make pretty good inferences about what's a feeding dive and what's not, just based on the shape of the whale's dive. So I didn't talk about dive shape, but whales will often conduct what we call a square-shaped dive. So they'll dive down, and then they'll swim along the bottom, and then head back up. And while they're swimming horizontally at maximum depth, they're believed to be ingesting prey. So they're diving down to a depth where they found a good prey resource. But there's other dive types, V-shaped dives, where the whale will dive down deep and then head right back up. So they're not spending much time at maximum depth. And those V-shaped dives are thought to reflect exploration. They're searching for a viable prey patch. So just by looking at the shape of the dive, we can infer what's a feeding dive from exploration dive. And then in terms of figuring out, prey densities? Forget the second part of the question.
Yeah, how deep? How do they know how deep to go?
Well, how do they know how deep to go? I don't know. A lot of people want to know how whales are able to find their prey. It's a really good question. There's various hypotheses about whether whales can smell their prey, whether they're communicating with other whales that have found a good prey patch. Understanding how animals interact with their environment and how they find their food is a complex question that I don't have a straightforward answer to at the moment.
Okay, and there's actually one more final question. This one is coming from Isabella, and she's asking, is there regulated whaling where you did the, and how does that work?
Yeah, also a great question. So bowhead whales, just recently, subsistence hunting has been opened up. And for this population, there's around 3 animals that are harvested annually from the territory of Nunavut. It is highly regulated. There is a veterinarian who actually participates in the hunt to make sure that it's done humanely, to make sure that all of the animal is being utilized. So it is something that is highly regulated, and as being someone who's witnessed the preparing of the animal and the flensing and everything, I can say that it does have a lot of cultural significance to the community, that the animal is completely used. I was very impressed with the utility of the animal because the meat is provided to the community. The baleen is provided to artists who are making jewelry. Same with the bones as well, are sent to artists that are making sculptures. So the entire animal is utilized, and it does provide a lot of cultural significance to the community. Yeah.
Oh.
Normally, no one asks bowhead questions. This is so great.
Oh, bowheads are fascinating.
They are.
I was wondering, from the blubber samples, will they also be looked at for toxins, seeing as that seems to be a growing problem in the Arctic?
Yeah, so the samples that we collected, many of them are going to Fisheries and Oceans Canada, and I am imagining that they are going to look at toxins as well. Which ones they're looking for, I can't say for sure, but yeah.
And I'm going to ask you one last one, which is, can you just comment a bit about the relationship between scientists coming from southern communities, working with the Inuit, and how you're doing outreach or how they're interacting with scientists?
Yeah, no, also a great question. So we've been very fortunate that the community of Pangatung has been very welcoming of us and of the work that we've been doing in collaboration with the community. So for the past 4 years, we've been working with the Killabuck family. They are expert hunters, boaters, fishermen, and they provide a lot of local knowledge about the environment and also about the whales that we're studying. And often while we're in Pangertung, the community will be radioing us, letting us know, oh, we found some bowhead whales, or oh, we actually found a group of killer whales that were harassing bowhead whales. So they provide all kinds of interesting insights and helpful information that we can use to inform our research. We also enjoy having community meetings. So we go to the Hunter and Trappers Association, we sit down, we talk with hunters, we talk with community members about the work we're doing, about observations that they see in their own backyard, and it has been overall just a really wonderful experience.
Okay, thanks very much, Sarah.
Thanks.
And next we've got Cassandra Girdlestone. From belugas to bats.
So, anthropogenic noise in the oceans is on the rise, and in order to understand how that's affecting marine mammals, one of the tools we can use is a frequency map of the cochlea, which is a small structure in the inner ear of whales and all mammals, all animals. So what a frequency map is a species-specific map of the cochlea which correlates the frequencies with a particular location where they're detected. So as you can see here, we have low frequencies which are detected at the innermost point of the cochlea at the apex, and as we move through the cochlea, we reach the base where high frequencies are detected. So if we have a frequency map in place, for instance, if we were to find acoustic trauma or damage here, knowing what frequency is detected there, we could potentially determine the sound that caused that damage, and in turn, the source of that sound. And we can in turn do this throughout the cochlea. Now, how can we create something like this for a beluga? We actually have to use terrestrial mammals as a proxy. So here we have the cochlea again, and if we were to uncoil the cochlea and flatten it out, you would see something like this schematic here, which is representative of the guinea pig. And this is actually known as the organ of Corti. And you see that the top row is known as the inner hair cells, or IHC, and below that are 3 rows of outer hair cells, or OHC. Now, what you're looking at are actually called stereocilia, and they're rod or bristle-like structures that act as transducers to convert sound waves into electrical impulses, which are taken to the brain. Now, the width of the organ of Corti is the distance here between the inner hair cell to the 3rd row of outer hair cells. And in guinea pigs, it's actually the same size as it is in beluga whales. So as we move from the apex, which you see on your left, along the cochlea to the base on your right, there are morphological and morphometric changes in those cells, such as the cell width, the spacing between the cells, as well as the stereocilia length. And those changes can all be correlated to different frequencies detected at the various locations. So here I'm showing you the frequency range of hearing for the guinea pig, followed by that of the beluga. And you can see there's only a small area of overlap between the two, which leaves us with a large area where there's no data for the beluga. So to overcome this and fill in some of the gaps, we're comparing them to Parnell's mustache bat, because they, as you can see, have a similar frequency range to the belugas, and they both use echolocation for hearing. At this time, the guinea pigs are the only species for which there's a morphometric map, but in bats, the Parnell's mustache bat, there is a frequency map that's been well documented already. So they don't have the morphometric map, but if we can correlate the frequency map with those morphometrics, we can potentially extrapolate that to create a beluga frequency map. So these are the locations I've looked at in the beluga so far. There's 21 different locations we've looked at, and we've taken measurements of 25 different parameters at each of these locations. These are some of the parameters that we've looked at. So the distance between the inner hair cells to the 3rd row of outer hair cells, also cell width, the cell spacing, and angles, and so on. So I'm just going to tell you a bit more about 2 parameters specifically. So here we have the distance from the inner hair cell to the third row of outer hair cells. And on the vertical axis on your left, you've got that distance versus the horizontal axis, which is apex on your left, again, moving across to the base on your right. And you can see there's a decreasing trend in that distance as we move from the apex to the base. If we overlay the Beluga data, you can see there's a comparable trend for them as well. But please take note, specifically, the most comparability is among the higher frequency range. Now, if we look at the width of the cells that are located in that outer hair cell 1 row, closest to the inner hair cells, you see the cell width on the vertical axis. And then again, if you compare that to the distance from the apex, you can see that there's somewhat of a trend that those cells are increasing in width as we move from the apex to the base. And here is the Beluga data. So you can see again, the trends are fairly similar, with the most correlation happening in the higher frequency ranges. Another thing I've been looking at is the length of the stereocilia in the outer hair cells. So here's what we found so far for the Beluga. You can see it's quite a clear trend, decreasing as we move towards the base. And this is the bat data that we have so far. And again, the most correlation is happening in the higher frequency range. So this is all very encouraging. And again, if we can correlate those morphometrics with the frequency maps that already exist, we may be able to use this as a proxy to help create a Beluga frequency map. Thank you to these people who've made this work possible. And thank you to you for your attention.
Do we have any questions? Here we go.
Thanks. Thanks for your talk. I was just interested, how do you measure damage in like the carclia?
So that's something that I haven't had the opportunity to do yet, but if we go back, sorry. So these are what the healthy cells look like. So they're called sort of smiles or U-shaped. So that's an intact cell. So you can see that this is an intact cell. So if there was damage, it happens very quickly, and those stereocilia actually just disappear, and a scar forms. So you would see a blank spot where that cell should have been. And they can't regenerate in mammals.
So those are happy ears.
Those are happy ears.
So when you finally get your frequency map plotted out, so how does that get used to inform us about the effects of noise on Beluga?
Well, if we can create a working frequency map, that will allow us, when we do find damage, if we know what frequency caused that damage, so when we find a blank cell, knowing the frequency detected there, we could determine what frequency of sound caused it, and potentially then what was the source of that sound. So if it was seismic activity or something like that, or maybe not even anthropogenic sound.
So that next step would be to recover dead belugas, maybe in the St. Lawrence, maybe up in the Arctic, and then you would then look at the ears, compare it,
That's
to
right.
know whether or not they had hearing damage and potentially the source of that.
Exactly. So if we can figure out what a healthy baseline looks like, then we'll know what a damaged ear looks like.
Okay, we have two more online questions. One is coming from Beatrice, and she says maybe she missed it, but how many samples did you study? And she wants to know whether those are from Aboriginal catches.
Good question. So the beluga data that I've shown is actually only one beluga. The bats, we've used 6 so far, and the beluga data will be compared to additional samples which we have that are part of a larger study. And yes, the belugas have been obtained through subsistence hunting, which is legalized in the Arctic. The only way we can obtain samples for this is opportunistically, so either through a stranding, in which case the material would be degraded and less reliable, or through the subsistence hunting, which allows for a fresher, more reliable sample and more reliable results in turn.
Okay, and then we have one more question from Leonardo again. He wants to know, can a frequency map for beluga whales also be used for other cetaceans that use echolocation? And is the cochlea of all cetaceans that echolocate similar?
They're similar but different. So different enough that we would have to do this work for each species. It is a species-specific map. There would be expected to be more correlation between echolocating animals than non-echolocating animals. That answers the question.
Do we have any other questions from the audience? None? Okay, thanks very much, Cassandra. And our last speaker in this session is Lance Baird-Leonard from the Vancouver Aquarium.
Andrew, thanks everyone for coming today. It's been as Andrew mentioned, it's been a pretty tough day. It is a tough day at the Vancouver Aquarium, and we'll talk about that a little bit more later on. But for my talk anyway. Okay. So what I'm going to do is provide a fairly quick update on, however, Andrew told me yesterday there's a little bit more time, so it'll be a little bit longer than I was originally intending, to talk about, to update you on our whale photogrammetry project. This is a project that I've been involved in for a few years, principally with colleagues John Durbin and Holly Fehrenbach at the Southwest Fisheries Center in San Diego. The work, the study, this aerial photogrammetry study, this way of measuring particularly killer whales from the air, from aerial photographs taken with a drone, was something that John and Holly developed in the prior to 2010, really 2007, 2008, using manned helicopters. It was very expensive, but it was the technique seemed to be very strong. And then there was a bilateral a panel convened and a series of workshops run around 2010 or 2011, 2012 to look at the potential impact of salmon fisheries on southern resident killer whales in particular. And it was a very interesting series of 4 workshops altogether. And one of the conclusions of the workshop was that the panel, the scientific panel that sort of deliberated almost like a jury situation, to scientists, managers, and both marine mammal managers and fisheries managers making their case. The panel recommended that, or reported that it thought that photogrammetry, this way of measuring the body shape and inferring the body condition of killer whales, provided was likely to yield the greatest sort of amount of new scientific information to shed on this question of how specifically how killer whale populations and killer whale individuals are affected by fluctuations in their prey supply. So armed with this recommendation from the panel, John and Holly and I struck up a little coalition and started a project to do this work using drones as opposed to manned helicopters because they were so much more affordable, and they turned out to have a number of other advantages too. They were much quieter and much less invasive, really, than photographing from a helicopter. Of course, individual variation in a bunch of killer whales, like as you see in this picture, there's a really thin whale up the top, a robust average animal of average condition in the middle, and a pregnant female at the bottom. Lots of things can affect body condition, and not just prey limitation, and these would be things like disease, for example, like age. We find that lactating females tend to be consistently a bit skinnier than other females. Not surprisingly, lactation is really expensive. And so part of the challenge is to sort of tease out this individual variation from sort of group variation that's likely to result from food effect. Killer whales share their food pretty compulsively, as Graham Ellis showed years ago now. So we think that when prey are limiting, that sort of prey shortage is likely to be more or less averaged over a group of killer whales. So thin group is much more likely to be indicative of a lack of food than a single thin individual, but very simply. I'm not going to talk about this much, but of course part of the intent of this project is eventually to link this better understanding of the relationship between food availability, prey abundance, if you like, and killer whale body condition to fisheries management. And the basic logic is outlined here, really. I can talk about that a little bit more in the question period if you'd like, but this is certainly where our intent is and our hope is that this is where the project will go, that this is really a study that we're looking at as an applied study. I'm going to I have a few slides to talk about pregnancy. It turns out that we can, as you could see from a couple of slides ago, it's when females that are highly gravid are pretty easy to detect in an aerial photo. We had the opportunity, John and Holly really before, as in the early stages of my involvement with the project, did some work at SeaWorld in San Diego with a young pregnant female named Kalia, and they were able to document using exactly the same kind of vertical photograph taken from a vertical perspective, changes in her shape and girth throughout the entire course of her pregnancy. They started a little photogrammetry study there before she was even known to be pregnant. And what they found was that that as you might expect, the width profile or the distribution of body mass, I guess, changed as through the course of Kalia's pregnancy, so that initially the point of maximum girth tends to be around the base of their ribs, and as pregnancy progresses, they get fatter down below. So we know what this is like. It's the same with people, so it's not just the fatness of a whale that tells you that they're pregnant. It's where that shape is, how that shape is distributed. Here's L91 a week prior to giving birth, and then after her calf was born. And you can see that bit of, that hasn't completely gone back to her sort of pre-gestation shape, but her shape in the right-hand side of this picture when she's showing with the calf is much less pear-shaped than in the left part. Okay. J17 was a tank. You can see she just wanted to have that calf so badly. But you didn't really have to do any science to be able to tell that she was pregnant. Here's a bunch of calves. We're also looking at growth rates. It's easy to measure length in these aerial photos. Interestingly, of these calves in the southern resident community, born in 2015, you can see that J50 is the smallest, and is also the oldest. Was the oldest when each one of these photos was taken, and she's the only female in this crop. And it does seem that females are a bit. We've known for many years there's sexual dimorphism. As adults, females don't get as long as males, about a meter shorter on average. But that size difference seems to begin at a fairly early stage, although it's pretty subtle to start with. A couple of, just to throw in some northern residents here, a couple of pics taken this summer of pregnant northern resident females. This is a picture of K27 carrying a fetus in May of this year, of 2016. We took this photo and didn't even realize at the time that she was carrying around a fetus. We thought it was a fish, and when we looked at the pictures that evening after taking this photo, we realized that in fact she'd had a sort of fairly late-term miscarriage. And one of the reasons, I guess, we're so interested in being able to detect pregnancy from the air is that there are multiple lines of evidence going back quite a long way that killer whales likely lose a lot of calves, either as sort of late-term miscarriages or as neonate mortality. So in tough years, it may be that it's not a problem for females getting pregnant, but it's difficult for them to carry those calves full term. So as far as this project goes, we started more or less as a pilot study in Johnson Strait in 2014. we've done Johnson Strait again in 2015 and 2016 at the same time of year, so that we can look on, so we have some consistency. We can untangle seasonal effects. We started southern resident killer whales in September of 2015, based on San Juan Island, working mostly in Harris Strait. And then we did that again in May of 2016 and September of 2016. So with southern residents, we're putting a bit more effort in, and we're trying to look at within season effects as well as between season effects. It'd be great to do that with northern residents too, but you know, our time and funding is a bit limited. We're focused, the study's really focused on resident killer whales, but we're also doing Biggs killer whales opportunistically. I didn't, oh yeah, humpbacks. I didn't put a picture of Biggs, of any Biggs in from the air, because that, you know, that's sort of a half-hour discussion. They're really cool. They're so different. They're big, jowly heads. They're like sculpins from the air. And every single Biggs killer whales we've looked at since 2014 has been fat. They're doing really well. So yes, in 2016 we started working on humpbacks a little bit as well. Our primary focus is on killer whales, but you know, humpbacks are very interesting as well. And of course, you know, we expect big seasonal conditions in humpbacks because they migrate and spend, most animals, and spend some time of the year in relatively unproductive calving grounds where they're not feeding much at all. I just threw up a few pictures of some of the well-known humpbacks from the northern Vancouver Island area, and Jackie is not in the audience, I don't think. No. Oh well, Jackie Hildering, Krista McMillan really helped a lot with the and Jared Towers with the humpback work that we've been doing. We've been using their catalog to match all of our humpback whale photos from the air. Humpbacks are really easy compared to killer whales for this kind of thing. Here's Slash. Those of you who spend any time up there are familiar with this whale. Here's her calf. Maud is a really big humpback female that's been coming to that area for many years, 1993, and she has had a calf this year, 28 feet long already for calves, pretty big. And here's some miscellaneous photos of some of the humpbacks. I threw this up because you can see the variation in body condition, and you can see Jackie's in good condition down there as well. So I just wanted to show this little video. We've also been doing, do we have some audio, Rob, by any chance? That's not super important. We also got a permit at the last minute to do humpback whale blow sampling, where we're flying the drone through the blow of humpbacks to obtain whale snot, basically, to and that's being used in two studies. One by our friendly local pet veterinary pathologist Stephen Rafferty, who's really interested in what kinds of organisms healthy whales have in their blow, and we're splitting the samples, and some of it's going to Michael Moore at Woods Hole Institute, who's looking at microbiome communities of microorganisms in the respiratory tract of humpback whales. If you could hear the audio here, you'd hear the way we do the drone work is that John does the flying. And Holly sits there looking at a screen, telling him what to do. So she's saying, back, forward, and he's saying, slow down. And it's always dialogue. I just drive the boat and listen to conversations. But anyway, this has been quite successful too. The humpbacks we haven't been able to detect any reaction on their part. We don't have any reason to think that they even recognize the drone as something other than, as something interesting. They have, of course, when they're feeding, there are often lots of birds around them, and so they're presumably used to flying objects. And we have you could sort of see maybe a little bit of a frame on the front of the drone. In theory, the blow comes up and coats that, and we throw it bag it up and label it and put it in a liquid nitrogen dewar. In actual fact, the drone ends up all slimy and rusty, and we swab it down and sterilize it, and we get lots of blows. So we didn't invent this method. Michael's been using it for a couple of years. Others have been using it in a study in Australia, but it's an effective technique. Thank you very much.
And time for a few questions before our break.
Would this change the whole photo ID program from one right side of dorsal fins to looking at photogrammetry? So you're looking at the whole animal?
Yeah, it's a good question. Do I need to repeat the question for the... No, you've got a mic. Okay. Does it change the whole photogrammetry program? I don't think it will really. I mean, I wouldn't like to imagine a world where everybody's driving around following whales and flying drones and crashing into each other and and into whales and stuff. I mean, I think there is a opportunity for mayhem here. But yes, we do get good quality pics, as you can see, and it was more easy than I think any of us expected initially to go from an aerial image to a horizontal catalog image. We haven't had any particular difficulty identifying the whales from the air. There are lots of things that you do get from the aerial photos that are more difficult to get from a boat, to do with some of the stuff that Sarah talked about. I think made that very clear. But it's particularly useful for seeing any injuries. And we've seen now 3 examples of killer whales that have obviously been struck and had significant wounds from boats, and 2 of those were unknown prior to us seeing that from the air. With humpback whales, we can see entanglement scars and so on them very well from this vertical. But no, I don't think it'll replace.
Another question over here.
I hope it doesn't.
Hi. I actually spoke to Michael Moore about this a couple months ago, but I want to know what you think because I'm hoping you have a different answer. I'm very interested in measuring acute stress response in particularly southern residents. I'm a prospective graduate student, and I spoke to him about potentially using drone technology to get blow samples from these guys, and his response was that their blow volume is probably not enough, and that getting the blow samples from humpbacks is difficult enough. And I was wondering what you think about that, if you could collect samples and measure glucocorticoid levels.
Boy, I'm not an endocrinologist, but we do get a fair bit of material. I mean, Michael's been doing this for a long time. He's very good, so I wouldn't contradict him. But what so here's something that might be relevant, I guess. In the last few weeks, in fact, Michael has found that he can do everything he needs in terms of the cultures from the microbiome work from a very small amount of material. So we gave him some samples that we thought were hopeless because we really didn't get much. And he got something from every single one of them. So that means there may be more of the samples left over for that kind of thing. And I know there's a group at NIMS that's very interested in the same question, and we can talk about that if you like afterwards.
We are going to take a 15-minute break. We're going to start right again at 1:15. Would all the speakers please come down to the front in case there are people who like to find you and chat with you? And thank you very much.
Much more relaxed this year.
people are sitting down and if we can get just a little bit of quiet. Our next speaker is going to be Valeria Vergara, but before she starts speaking, I still need people to be quiet. Let's dim the lights one more further down. And we'll make it more dramatic. There we go.
But I just turn it off.
No, that works. No, well, just for the moment till we get the people sitting down. The quiet is down. I just want to say a couple things before we start up again. The first is just to acknowledge our sponsors. This has been very much a grassroots symposium, and we have done it with a lot of volunteer help, and we haven't charged anybody anything to cover some of our basic costs. And we're therefore grateful for the financial contributions from the Pacific Wildlife Foundation and from the Pacific Whale Watch Association, as well as from UBC's Marine Mammal Research Unit. I just got a report from Marcus in terms of the number of people that are participating online. We just hit 810. One of the people apparently commented, it's great having access to the University of the World. And so I think we're onto something here. Our next speaker is Valeria Vergara, and will speaking to us on belugas.
Hi, everybody. So I want to talk to you today not so much about results, but about the pairing of two technologies that for people that are, for biologists that have been interested in animal communication for a long time, like me, is really advancing the way we understand what the communication between these animals is all about. After years of spending time describing behaviors such as milling or traveling or directional, all of a sudden we can see the detail of their interactions from the air while we record them underwater. And this is really, really groundbreaking for cetacean research, and in my case, for beluga research. The first example of this is all these pictures were taken in the high Arctic at the north end of Cunningham Inlet, where I spent 2014 and 2015 conducting a study of beluga communication in the absence of anthropogenic noise. And there were 14 beautiful instances of natural entrapments where the groups of whales would get cut off from the larger aggregations and then reunited after a few hours. And we had access to a small drone for a few of these entrapments. And so we were able to tell with much precision the group composition of these groups. So not only the number of adults, juveniles, and yearlings, but also which groups had calves. And because when whales are entrapped, they produce contact calls a lot more often than when they are just swimming around in the delta. And my study has focused on contact calls since I started trying to understand the communication of these animals in 2002. This was really the perfect opportunity to try and further our understanding of the function of these calls. So what can you understand when you have a visual from the air paired with acoustics? You can look at the proportion of contact calls in relation to, and other call types in relation to the number of young animals in a group, the numbers of calves and yearlings. You can look at the number of contact calls in relation to the number of adults in a group, and that tells you a lot about signature calls, and I will present this incredible data next year, I promise. And you can also look at calls, whether there are calls in groups with newborns that do not exist in groups without newborns, and this is very illuminating as well. Fast forward a year, 2016 was the first season of my study in a population that has now been formally declared endangered by SARA. This is the St. Lawrence beluga population. Calves in the St. Lawrence have been dying at unprecedented numbers in the past few years. The St. Lawrence is a very noisy place. This is just a cross-section of ambient noise from the mouth of the Saguenay, where there is a ferry lane. And we're looking at whether how mother-calf acoustic contact is compromised by noise. And again, pairing our digital hydrophones with visuals from the drone has been invaluable. Because we can look at the number, the type, and the location of vessels in relation to the whales from the air. We can also really clearly look at group composition and target those groups that have young calves. The younger, the better. And we can also tally the number, all the occurrences of mother-calf separations. And we can ask and attempt to answer, of course, things like, do these whales use contact calls when they are apart? Are these calls masked in noise? Under what levels of noise are these calls masked? Do the groups come closer together when in situations of noise? Do mothers and calves even swim apart in noisy areas? Maybe the mothers don't want to leave the calves or vice versa if it's too noisy, because regaining contact might presumably be a little trickier. So this is an example. It's a little quiet, but you can hear some of the calls. You can. So the characteristic of the St. Lawrence is that it's very turbid. I would doubt that these animals can really see each other when they are a couple of meters away from one another. They need to hear each other. You can see a little calf at the head of the group. The animals are immediately oriented towards the little calf. You can hear a few faint contact calls. So this is the kind of thing that we're looking at. The next, I'm just going to let you absorb this and then I'll talk. So this is a group of males. We knew that we had with us a group of 15 males because a few of them had been consistently photo ID'd. Extremely social scene. These males were incredibly tactile with one another before, during, and after this scene. We videoed them for about 20 minutes. They acted very much like baboons in that they would gang up against the deformed one, whose name was Neo. You probably saw one of the whales had a little bit of a scoliosis. They would gang up on him. You would hear aggressive sounds, and then they would come together and rub with each other and engage in this incredible affiliative behavior, much like the fights and reconciliations that bonobos, not baboons, have. At least it was reminiscent of that. And another interesting thing of this scenario is that we knew we didn't have any females around, we didn't have any calves around. It was a dead-end sort of a bay based in Marguerite. So understanding what kinds of calls you have in the absence of mothers and calves is important. And another really, really interesting thing of being able to pair these two technologies and do photo ID at the same time is that when you hear the same, like the trumpet call, you all could recognize it, I'm sure. It was very, very distinct. We heard it again when we had Neo hanging out with a group of females and calves. Now, we can't be, and that was the only call that there was in common. We need many more of these events to start, matching calls to whales, but it's just telling. It's just one of the little things that we can begin to start to find out by being able simply to watch things that we couldn't before. So thank you to a really great research team and many other associations. But most importantly today, I want to thank Aurora and Kila. without them, this trajectory that I've been on since 2002 would have been impossible. So I'm very, very grateful.
Do we have any questions? Okay.
When you say entrapments, what are they entrapped by? Ice or?
No, great question. So they would swim up narrow river canals, and the tides are very fluctuating, and so the tide would go down, and they would get entrapped by sandbanks, and literally like in a pool. And then as the tide would go up, they would be able to get out.
Just a really quick question. What happened to the little calf that was out in front of his group, getting farther and farther away?
Oh, they caught up to him. No, a good ending.
Okay, thank you. Megan Moore.
Okay. Thank you, everyone. I am actually standing in for Caitlin Birdsall today because she is still up in Prince Rupert. And so I am here to share a new study that Caitlin and Lance have started looking at seasonal and daily patterns of habitat use by harbor porpoise in the Prince Rupert area. Sorry? There we go. So harbor porpoise are widespread in nearshore coastal areas. However, studies have shown that individuals may be relatively restricted to certain habitat in their habitat range. And small areas characterized by specific habitat features may actually be used more frequently and are thus quite important. So this low dispersal and targeted habitat use may actually make harbor porpoise vulnerable to local depletions. So in 2014, Caitlin moved north and helped establish the Vancouver Aquarium North Coast field office in Prince Rupert. And the Prince Rupert harbor, although it's quite a busy industrial port, it's actually an area where harbor porpoise have been quite commonly encountered. And since being up there and conducting surveys, Vancouver Aquarium staff have encountered harbor porpoise in that area around the Prince Rupert Harbor every single survey throughout all the seasons. Also interestingly, I think there we go. This is information from a map from the BC Cetacean Sightings Network database. That also shows that harbor porpoise are the most commonly encountered cetacean in the Prince Rupert Harbor area. And this red circle is identifying a cluster of encounters on the map that is from the database. And that is in the same location where the aquarium staff were encountering the harbor porpoise the most during their surveys. Also interestingly, during some of these surveys, Vancouver Aquarium staff were seeing large aggregations of harbor porpoises. And so these large aggregations are 50 or more animals, and they encountered these mostly in February and January in the wintertime. Also to note, in the BC Cetacean Sightings Network database, they have 5 high-confidence sightings of large aggregations of harbor porpoises in the same area. So it was something of interest that really sparked their attention and they wanted to look at more. Also something unique about this area was they were starting to notice behavior in the harbor porpoise that wasn't normally or typically seen with harbor porpoise. And so this was high energy behavior, so fast swimming, porpoising, or rooster tails that are more commonly recognized with Dall's porpoise. They were seeing harbor porpoise approaching boats and wake riding. They were also seeing what they thought was potentially mating behavior, and so that was recognized by 2 individuals fast swimming together, breaching, and genitalia was present. And then they were also noticing spy hopping and leaping in harbor porpoise. So with all of this information, they wanted to start a pilot study, Lance and Caitlin, in July of 2016 to start looking at the seasonal and daily patterns and habitat use of the harbor porpoise, specifically in the Prince Rupert area. They were looking at changes in habitat use associated with anthropogenic factors, interested in changes in group size throughout the year, and, if possible, comparing the information from the Prince Rupert area to other areas. Should mention, forgot to in the other slide, that the area in Prince Rupert Harbor is, from the Sightings Network database, is the second highest amount of sightings, of large aggregations of sightings in one area, second to the Strait of Juan de Fuca Strait, which Dr. Anna Hall identified and described earlier. So that's something that they would like to compare to. So to do this study, it's a two-part method. They're using an automated click detector called a C-pod that's deployed and anchored underwater in the harbor to detect the clicks of harbor porpoises. And then they're pairing that with land-based observation studies. And that's to ground truth what the C-pod is detecting and to record information about the different harbor porpoise and vessel traffic. So these C-pod detectors have been used quite regularly in North America and Europe to detect harbor porpoise clicks. It's detecting the high-frequency clicks of harbor porpoise, and recording these detections as a text file. The land-based studies is using a similar method to what Aileen Jeffries set up with the Pacific Biodiversity Institute in Washington. So they're doing counts of harbor porpoise on 10-minute intervals in the grid area of the study, and they're recording the harbor porpoise behavior, as well as the direction of travel and anything that they're seeing they're doing. They're also recording vessel traffic in the area. So the vessels that are transiting through, they're grouping the vessels into type and recording their time that it takes to transport through the area, recording what they're doing, whether they're fishing, just transiting, other work. So this is just the beginning of this study. As we noted, the C-pod was just deployed in July. Sorry, you threw me off, Andrew. This is my last slide. In 2 months that it was down, it's been collected once, and they had over 6,000 detection positive minutes for narrowband high-frequency clicks. Now, because the Prince Rupert Harbor has very high anthropogenic noise, they do have to go in and verify each detection to make sure it was a harbor porpoise and not some other noise factor. So that is what they're in the process of doing right now. Just a quick thank you to Aileen Jeffries for her help and knowledge in setting up the study, James Pilkington and Jared Towers for their help and suggestions for deploying and retrieving the C-pod, and then Prince Rupert Port Authority and Nexon for help getting to the C-pod in the site.
Do you have any questions for Megan? Gotcha. Where were we? Raincoast. Brian Faulkner.
Thank you, and thank you, Andrew, for an opportunity to once again do a completely blatant commercial for both Raincoast. and for our marine operations program. For those of you who don't know, Raincoast Conservation Foundation is a nonprofit, science-based organization. We are a registered charity in Canada. We conduct some of our own field research, as well as we have a lab at the University of Victoria, the Hakai Raincoast Applied Conservation Lab. So my role at Raincoast, one of them, it's a nonprofit world, so you get 2 or 3 titles and half a wage. But it has been to develop the marine capacity to work in the areas where we've worked for the last 25 years, primarily in the Central Coast, in the Great Bear Rainforest, and in the offshore waters around Haida Gwaii and Hecate Strait. So this is our primary research vessel, Achiever. Some of you, I've had the great pleasure of having a number of you on board as researchers. We've done a lot of different marine mammal work, usually in conjunction with the Biological Station in Nanaimo, and with IOS, we've done basic oceanographic work. So we bought this boat in 2003. We did a massive refit on it. We've used it for about 10 years, done a whole variety of different research, some of it our own and some of it in conjunction with Andrew, some of the sea lion censuses that we've done over the years. It's worked extremely well. This year, after 10 years, we did another major refit on it. It was a major expenditure again, and yet it's newer than ever, and we've upgraded some of the electronics as well. So it's in absolutely brand new, pristine shape again. So we're looking forward to another 10 years. The fellow who should be doing this presentation now is a gentleman named Nick Sinclair. He's taking this project over from me, so he gets now a quarter of a job, or a quarter of a wage, and is taking this project over. So I'm focusing on some of the other jobs that I have to do. But for the last 10 years, this has been an enormous part of my life, and it's been a huge privilege to be engaged in some of the work, the research work that we've done. So the idea when we got the boat was to build something that was completely practical, very highly adaptable, so it has lots and lots of workspace and lots of accommodation. So we can sleep up to 12 people, we can turn that into work tables, and they can sleep on the tables at night. So it's a really adaptable boat with lots of accommodation. We want to weld things onto it at any point. If anybody has any equipment or anything that they need to weld on it, this is an observation platform that we've used for basically all 13 of those years, doing marine mammal cetacean surveys and other projects with it as well. We've done a series of our own marine mammal surveys, which was done over a 5-year period, beginning back in the days when people were talking about pipelines as if they were sort of these mythical things before the word Enbridge became even known. And we conducted those surveys for a number of years and produced an awful lot of material, both published material and then some of that published material converted to popular publications, which have really gone a long ways to informing the public about much of the value of the distribution and the abundance of these marine mammals, but also the potential risks. Sadly, our own lab, for me, is focused an awful lot on terrestrial and has been over the last number of years. We have one of the most amazing and largest grizzly bear and top carnivores that depend on salmon. It's a project monitoring them over the territories of 5 coastal First Nations. So it's an enormous project over a huge area of the coast. But unfortunately, most of it's in the forest and up in the mountains, so we don't get a lot of work from that. So, one of the things that's really important in our relationship and the work that we do, both on the ground and on the water, is a partnership with First Nations. One of the speakers was talking about how valuable that relationship is, how it's informed the science. In fact, it's leading the science that we do in a lot of cases. So the relationships that we've developed over the years with those First Nations on the coast, in the coastal areas, has been incredibly positive and extremely productive. So as I said, this is probably the closest we get to doing most of our own marine mammal work at this point. We have a couple of grad students in the lab now that are working on marine projects from existing data. But I guess what I'm here to do is to essentially a commercial for this. We have not only the marine capacity with Achiever, she has 2,000, 2,500 mile range, tremendous water capacity. We can be out for 10 days, 2 weeks at a time. Extremely efficient platform to do marine research from. So if you have any ideas or projects, the second thing that we sometimes have access to is magic word funding. Being a charity, we have the ability to help people with tax problems. So if any of you made so much money this year you have a tax problem, we'd be very happy to help you out. But we also do have a donor base, and some of those are foundations, and many of those support science work. And some of them are private donors that have individual interests in some of these things. So if you have any interest, any projects in mind, by all means, come to us, and we'll get you out on the water. Thank you, Andrew.
Thanks, Brian. Any questions for Brian? I might just make two comments. One, when I saw the grizzly bears up there, I was reminded that one year we had somebody come and make a presentation to make the case that we should add another marine mammal to the list, which was the Sasquatch. I'm sure some of you remember that presentation. But I also know that on this coast, we have very few vessels to use to take groups of people out. And from a university perspective, we want to be able to take students out. So that's one boat that's well set up, and Pacific Wildlife Foundation has another boat. And we don't have enough of them in BC. And to have boats and skippers that understand the need for research and how it can be done. So the Achiever is wonderful. I've used it, and so have many others here. It's a platform. I should mention for people who are watching online who don't have the benefit of the agenda that was handed out, that we wrapped up our session on whales, dolphins, and porpoises. We're now into the session on research programs. And our next speaker is going to be Anna Hall, talking about going global. And as Anna's coming down, again, for those online, some of the best talks are coming this afternoon on seals and sea lions. Stay tuned for after lunch.
Thank you, Andrew. Whilst perhaps the talks on seals and sea lions are this afternoon, I have the pleasure of talking to you about what I believe is the world's greatest small cetaceans, the porpoises. And for those of you who know me, you'll know that I've been working on these animals for quite some time. And during the course of my graduate research, there were a number of challenges that kept cropping up. And they weren't specific to me. It was for people working on porpoises around the world and meeting people at conferences, and we would all have the same complaints that access to the literature on porpoises is very, very difficult, and unless you happen to be at a big university like I was at University of British Columbia. Having communication between researchers, even just being able to find out who else is working on these very small, very difficult to find, and geographically usually disjunct species. So after finishing here at UBC, and continuing on with research, it came to light that what we really, really needed, what was missing on planet Earth, was in fact an organization that was dedicated to the research and conservation of porpoises. It was a gaping hole. And luckily, I was fortunate enough to be involved with a group of people who were like-minded, and we formed the Porpoise Conservation Society. And we wanted to think big. For the world's littlest cetaceans, we wanted to think global. So it took us a bit of time and a bit of effort and deciding what are the ways that we can really help these animals and what do we need. So first of all, of course, you have to have a website. You just have to. And so we have the only website that I know of that is dedicated to the world's 7 species of porpoise. And I'm going to tell you a little bit about what you can find there, and I hope it's helpful, and I hope you may visit it. The mission of the Conservation Society is that we work for the protection and conservation of all of the species of porpoise on this planet and their natural habitats, and we do so through outreach, through education, and through research. Now, the Porpoise Conservation Society has a local base. It's based right here in southern British Columbia, but we have an international scope. And as I've mentioned, we are the only group on the planet that is focusing entirely on these species. And this is a Dall's porpoise. It's a fantastic photograph that was contributed to the Society. And you can find us and some of these photographs at porpoise.org. And our scope, as I've mentioned, is all 7 species, from the beautiful Burmeister's porpoise of the Southern Hemisphere to the finless porpoise of the Indo-Pacific. And one of the things that we had realized was that when you go and you try and find what are the distributions of these 7 species, for even animals that are in, say, British Columbia, there's really very little fine-scale data. There's little snippets here and there, but by and large, if you look at the range of any of these species, there's huge swaths of ocean, usually coastal with the exception of spectacle and Dall's porpoise, in big bands. And yet, from experience with harbor and Dall's porpoise, we know that's not true. There is fine-scale variation. And so the Porpoise Conservation Society developed a global reporting platform that's available online, and anybody can report their porpoise sightings. And the goal with this is to eventually be able to pinpoint some of these hotspots, as with the work I did here in British Columbia, the areas that are incredibly important to the different species, so that we can help refine some of these maps, the habitat distribution, which of course is absolutely imperative for effective conservation. And our society is quite new. We've just had our first anniversary back in May, and we're getting sightings already from all over the world, and from places that we wouldn't have expected, that perhaps wouldn't have heard about us. For instance, in the UK, we're getting quite a few sightings from their sightings reports. And so our database is growing, and we're able to start pinpointing certain locations just at the very early stages of the reporting platform of sites that perhaps for graduate students or researchers might be areas that would be important and important habitats to start looking at. If you see a porpoise, of course, please recite it to the Sightings Network here through the Vancouver Aquarium, but please also report it to porpoise.org. There it is again, just in case. Now, it is very important, of course, to recognize that these animals are small, they're difficult to find, and we need money. And we just recently were granted charitable status through the Canada Revenue Agency, which is fantastic news. Our international work has been focusing on vaquita, which of course is the most critically endangered cetacean on the planet, with a current population estimate of 53 animals and declining. The reason for this is bycatch in illegal fisheries. This year we participated in the International Day to Save the Vaquita, which was on July the 9th, and we co-hosted an event with the Vancouver Aquarium here in Vancouver. And we were one of over 30 venues around the world, ranging from Canada to New Zealand. And the result of this international effort was Mexico implementing a full ban of gill nets throughout the full range of the vaquita. And since that is the single most important threat, this was a huge action. Our international collaboration with this species ranges from young children in preschool to fisheries, and we are now combining forces with on-the-ground efforts in the Sea of Cortez, or near the Sea of Cortez, and we have our first planning meeting next week as to what we can do for artisanal fisheries. We're also working with groups in Scotland and Wales, and we have a research program that's being set up in Scotland at one of the first special areas of conservation for harbour porpoise. That'll be commencing next year, and it's going to be building on 28 years worth of research that has already been conducted in Scotland. And we're also working on photo ID, because even though they're small, difficult to find, and incredibly difficult to photograph, we are finding that there are some individuals that can be monitored. And I had the pleasure of attending the first annual Porpoise Symposium, sitting in a room of people all dedicated to the conservation of porpoises. Fantastic. I also want to very quickly tell you about the Black Sea, because this has flown under the radar. The Black Sea population of harbour porpoise is actually a distinct subspecies. It's one of 3 around the world. They are listed as endangered by the IUCN. The coasts of Bulgaria and Turkey are known breeding grounds, and in this past summer, there was a mass mortality with over 300 neonates stranded along the shores of Turkey and Bulgaria. It's thought that their mothers were caught in bottom-set nets for turbot, and these animals starved to death and washed up on shore. This is not the first mass mortality, but this is the biggest one on record. We are working with colleagues at the World Bank and the IUCN to try and figure out what the Porpoise Conservation Society here in BC can do to help the people working along the Black Sea coasts of Turkey and Bulgaria. Our other resources, we have an online research library that has 818 documents, all about porpoises, various species, various researchers, and our library is growing, and it's available free online to anybody. We're also developing a photographic catalog from around the world of all the 7 species. And this is available to everybody. So for the students in the room, we hope it helps. So if we've got your interest, which I hope we do, please use our website. Please contribute any papers that you may have, photographs, and report your sightings. And of course, Christmas is coming. We are charitable. If you have somebody on your list who has absolutely everything, have they adopted a vaquita? We can help. We also have fantastic stocking stuffers. Our brochures are available, and they're free. This work could not have been done without a tremendous amount of hard work by some of the people in this room, and my incredible thanks to all of them and to you for your time. And even the universe knows small cetaceans are incredibly important. This was taken by NASA. It is the dolphin galaxy. I am positive there is a porpoise one out there somewhere. Thank you.
Okay, well, I think then we'll have a chance to catch up with you at the break. I had no idea. What was that website again, Anna? Excellent. Yeah. I mean, I've known Amana's passion for the porpoise for a long, long time. And it's really quite something to see, to take that passion and bring in an international community. It being done here in BC. So congratulations to you and the others on your team. Next we have Jevon Friedlander telling us a bit about the BC Cetacean Sightings Network.
Thank you, Andrew. My name is Jevon Friedlander. I've recently started as the program assistant at the BC Cetacean Sightings Network, working with Tessa Danilesco over there. We're running out of Vancouver Aquarium's Coastal Ocean Research Institute. So I'll just be giving you a brief update on what we've been working on the last year and some of the projects we have upcoming. So for those of you who don't know who we are a sightings network. We collect observer-based data. We collect these sightings on the 23 cetaceans and the 4 species of sea turtles that we find on the BC coast. We have over 5,000 unique observers that have contributed over 100,000 individual sightings reports, which you can see they're displayed on this map here. All of our data is available for collaboration in both a raw and a effort-corrected format. It's available for any research projects, any conservation assessments, or any environmental impact assessment, anything along those lines. So there's a number of ways you can actually report to us. You can report via, we have, of course, logbooks we distribute, phone number, email address, website, where you can submit an online form. And also, most recently, we have our Whale Report mobile app, which we released in June of 2015 last year. It was a huge success. We have almost 3,000 downloads of the app to date, and has resulted in an 80% year-over-year increase in sightings reported, which is great for us. And it's resulted in more real-time reports that we're getting, which is, has a couple implications. Number one being that if we get a report of a distressed or a deceased animal, we can pass that information directly to the BC Marine Mammal Response Network, and they can act on it in a timely manner. As well, if we get a sighting of interest, for example, a unique or a rare species, one that's in an interesting location, we can act on that, we can further investigate it, and see what it's about. So the other major project that we've been working on this year has been the Mariner's Guide to whales, dolphins, and porpoises of Western Canada. So we drew inspiration from the Mariner's Guide to whales of the North Coast, of North Atlantic, which was created for the East Coast. So we're partnered with the Port of Vancouver's ECHO program, as well as the Prince Rupert Port Authority. We collaborated with the DFO, as well as working with just a huge number of different mariner organizations from throughout the coast, for example, BC Coast Pilots and BC Ferries. And we really wanted to work with them to create this guide because they're going to be the ones that are using it, and it's for them. And the guide, what it is, it aims to educate, raise awareness among mariners for the exact species that we find on our coast of cetaceans and sea turtles, some of the threats, the vessel-related threats that these species face, and ways you can mitigate these threats. So yeah, I should mention it isn't quite released yet. It will be released in December of 2016. These hard copies will be distributed via the Port of Vancouver and the Prince Rupert Port Authority, and we're also going to have an electronic copy on our website that's available in full. So this is an example of what you will find in the Mariner's Guide. As you can see, it outlines the species, it identifies, it describes, it shows the conservation status. This is just the gray whale, an example I took from the book. And then on the right, you can see there that it shows these seasonal relative abundance maps. So a lot of data went into the making of these maps. We used our own Sightings network data. We used DFO. They had aerial and boat-based survey data and Stantec survey data from the North Coast. So we used all these data to create these relative abundance maps, which really show where you'd have a high probability of finding certain cetaceans in certain seasons all along the BC coast. So we have these for all the cetacean species you'd expect to find, and we also included these quick reference inserts. So the point of these is just to be a one-stop shop for all the information you would need. It goes right on the bridge of the ship. So you can really look at one glance and see where the hotspots for cetacean activities are in both the north and the south coast. And on the right there, if you continue, it has steps you can take to mitigate some of the threats that may be posed to these cetaceans, should you encounter one or should you know that there's one in the area. So that's what we have been working on. Some of the projects that are upcoming, we're going to be further exploring the utility of our real-time data. So we're getting all these data in real time now from our app reports, and now we're looking into utilizing it for a direct-to-vessel alert network, or a message system that will really be sent directly to the vessels of large vessel crew. If we get a sightings report that's in the area, that's in an area of high vessel activity, we can send that information to the Coast Guard, which can then relay it directly. Oh, sorry. We can send that information directly to the Coast Guard, which can then get sent to the vessels that are in the area. We're working to expand our volunteer program via coastwide Sightings Network ambassadors. So we'll be working with conservation leaders in different communities. We'll be training them and educating them to really act as Sightings Network liaisons. So they can, in doing so, we're hoping to add a more sort of dynamic organizational structure, which in turn hopefully will raise awareness in different parts and result in an increase in sightings reported. Another couple projects we're working on, an aerial identification guide. So this would be a reference for sea air pilots and their passengers, as this is a group that is rather underrepresented in our database. So we're hoping to give them the education and give them the training they would need to identify these cetaceans with accuracy and hopefully result in more sightings reported from that observer base. And finally, our cetacean census. This is further working on and honoring the work of Dr. Michael Biggs and his killer whale census. So we're going to be looking to launch that in August 2017, piloted with different eco-tour operators from around the coast, different mariner organizations, and boating clubs and groups. And that, in August 2017. And that is what the Sightings Network is up to.
So I presume there's something between those covers we could look at?
Yeah, there's a couple pages of information in here.
And can people look at it today and have a few copies available to look at?
This is the only copy I have, so please don't run away with it. But yes, I imagine they could
You'll be
take
here
a
at
look
the
if they wanted to.
Okay, at the end of the session, you'll be down here? And
I
you
will
can
be down here, as you mentioned.
Perfect. So much.
Thank you.
So the next two speakers on our list were unable to come. Lance Barrett-Leonard is going to speak to us about Aurora. It's going to come up next. Like right there.
Yeah, thanks very much, Andrew. So as has been mentioned a couple of times already, Aurora, our female killer whale, and as of a couple of weeks ago, our last killer whale at the Vancouver Aquarium. Was she a beluga? God. Oh man, this shows what kind of shape I'm in today. Yeah, passed away last night. So this is the first time in 49 years, apparently, that there has not been a beluga in Vancouver. Aurora's death was preceded 2 weeks ago by the death of her calf, Keela. I know there's a lot of questions about what could possibly have caused this double mortality. I can't speculate on that, and those of you who know me know that you can't really shut me up. And I would if I had any information. I really would, but I don't. Not only do I not have any information, but it's still very much an open question. Our vet, Marty Helena, Stephen Rafferty, the veterinary pathologist who's sort of world-renowned for his work on the pathology of animals in general, but he's certainly an expert on cetaceans, has not been able to determine the cause of death of Keela. Stephen and Marty are doing a necropsy on Aurora today out at the Animal Health Center. Really the range of possibilities extends all the way from sort of a fungal pathogen to a bacterial pathogen to a viral pathogen to a toxin. We're pulling the stops out, trying to figure that out. I got an email from Stephen this morning, and it's very much it's a very high-profile event. I think what I did want to say is Keela, the baby, the beluga passed away 2 weeks ago, was the baby beluga of the Raffi song. That inspired the Raffi song. Children throughout the Lower Mainland and throughout British Columbia have grown up with these belugas, young adults as well. The aquarium group, the aquarium family is devastated. I think a lot of people in the city are devastated. Right now, there's sort of an impromptu area of mourning at the Vancouver Aquarium, I guess, with the public are visiting, and the flowers and letters of condolence and so on are coming in. I guess what I'd like to say is that, from the perspective of someone who's worked at the aquarium, but gosh, I don't think you really have to work there to get it. No matter how you feel about marine mammals in captivity, you have to appreciate that being a trainer or being an animal care worker at the Vancouver Aquarium is something that most people who find themselves in those positions have aspired to for their whole lives. So, your kids that grow up in Vancouver, they want to be a marine biologist. There's at least one in the audience here today who has that aspiration. You work your brains out really to get the kind of position that would put you in contact with the animals at the aquarium. When you get there, you find out, if you're lucky enough to get a job as a trainer, you find out that you're at the bottom of the totem pole and you spend your time thawing fish, and if you're lucky, you start to get limited contact with the animals. When the kind of contact that people and cetaceans in particular form in these kinds of institutions is really it's quite amazing how strong that is. I mean, these animals really become, you know, as important or more important, I have to say, and sometimes as family members to these staff members. And for myself, I work in the, I've been at the aquarium for 15 years. I work in the research department. I was hired to work on wild, to conduct research in the wild. I've conducted a little bit at the aquarium, but not much. When I arrived, we had a very capable and competent and extensive program going on, run by UBC, by Andrew's group, working on the animals there. So my experience at the aquarium has been one really of interacting with those animals in a primarily a non-research capacity. When I started 15 years ago, my office was the underground bunker that some of you visited. My only window, it was the office that my predecessor John Ford had occupied. My only window looked into the beluga tank, into the med pool. So when I came in the morning, the belugas would crowd in the window to see me. When I left at the end of the day, if I did anything weird, the belugas were there to check me out. If I had a kid in the office, there could be 100 kids on the other side of the glass in the public galleries. If I had one child in my office, the belugas would all crowd to look in to make sure that kid was okay. My own son was 2 years old then. He resembled a beluga, I have to say. People would look at my wife, Kathy, and say, gosh, condolences, he had a big forehead. And he's a relatively long birth. He's 17 years old now, but he grew up with them. And, you know, whenever Lee was in my office, he liked to sit up in the window. You know, the belugas would all crowd around, and the trainers would eventually come down and say, what are you doing down there? Belugas won't come out for the show, you know, and because they were that interested. And I think it was on my first week working at the Vancouver Aquarium, I cycled into work early, standing there in my office with my pants around my ankles, changing out of my cycling clothes, and I look up and all the belugas are staring in at me. That's just, that's sort of how it was. And over the years, we've had, you know, we've had our ups and downs with marine mammals. We've had, but as everybody knows here, but the belugas have really been the mainstay of our marine mammal collection. And they're the, again, you know, no matter how you feel personally about marine mammals in captivity, you know, I defy anybody to contradict me on the reasons that we have them. We have them to serve as animal ambassadors, to inculcate a sort of a stewardship ethic and a care about these animals in the wild. That's why we do it. And you may think that's not the right way to do it, but that's why that's been our approach since day one. And I certainly feel that they've had that effect. You know, our feelings about marine animals in captivity generally evolve over time. I make a point when I walk through Stanley Park of walking by the old polar bear enclosure as often as I can. That's what we used to do. You know, I think our standard of care for the belugas was high, and it was certainly as high as we could make it. We did our best. I don't know. I'm happy to talk about, you know, where we might go from here, but I have nothing to add. I have no particular insight into that, I guess, that others here in the room wouldn't have as well. We're not sure what we're going to do. But I would ask, you know, for those of you who work at the aquarium or have worked at the aquarium or have visited the aquarium on a regular basis, you know, my sympathies to you at this point. And at the same time, I ask that you be somewhat sympathetic, at least, to the staff at this point. I find it very distasteful, Larry, I'm talking to you partly, when the press hammer us when these things happen. It's not fair. You know, the people who feel that, who take that criticism the hardest are the younger staff there. They're suffering from grief. When they pick up the newspaper and somebody's written an article about, you know, or flipped open their Rolodex and talked to the first animal, you know, no whales in captivity person that falls out, and to get a contrary opinion, and our staff have to deal with the grief and have to deal with the fact that they're suddenly pariahs, it makes me angry. And it's not a question of how you feel about it. about animals in captivity is how you feel about people. So give us a break for a little bit, please. And I'm happy to take any questions that you might have.
Anybody have a question? Thank you, Lance. And our last presentation before lunch is Lauren Hartling. You know what, I think we're going to take our lunch right now, and we will start up again at 1 o'clock. We have 45 minutes, and Lauren will be our first speaker. See you back here in 45 minutes. And we're ready to start again. So welcome back to the afternoon portion of our symposium. Mark has informed me that the numbers of viewers has gone up to just over 1,000. So I guess I'm now talking to you through this little camera. And we appreciate we're receiving some feedback. Some of you have requested that we have the agenda posted, and we will certainly do that for next year. It's our first time doing this, so we will be modifying it. So we do appreciate the feedback that you're sending in, and we're listening. Our first speaker, before we get into the seals, sea lions, and fur seals, is Lauren Hartling.
Hello, I am Lauren Hartling. I'm the interpretive specialist for marine mammals at the Vancouver Aquarium. We don't often consider ourselves researchers. We're interpreters. We get paid to talk, and we're very, very good at it. But one of the goals I had for myself this year was to do a bit of research, specifically on the calls to action that we do in our interpretive programs. So there you go. You can start off with an adorable picture of a California sea lion. The one that's marked. Okay, cool. So maybe if you've never heard the term call to action before, just so we're all on the same page, in terms of how we use it at the Vancouver Aquarium, a call to action is an action that our guests who are watching that program can take in their daily lives. So perhaps it's the first time they've ever seen a sea otter, a very cute baby sea otter, which we now have 3 of. Maybe you want to find out how you yourself can help protect sea otters. Maybe it's something to do with a snake. Maybe you've always been terrified of snakes, but by coming to the snake program, you discover, okay, cool, they're actually not scary, they're important, I shouldn't be afraid of these creatures. We do have call to actions in basically every interpretive program at the Vancouver Aquarium. One of my favorites is in our 4D theater, one place you don't often expect to see it, for the porpoise.org group out there. This is Levi, the rescued harbor porpoise we took care of at the Vancouver Aquarium back in 2013. So at the front of the 4D film, you actually have this snippet of his rescue, rehabilitation, and release. So it's a really great opportunity to share one of those positive stories with people in a place where they actually can't run away from us. Also, the things you don't know. There was Oz! You guys are great. Yeah, that is an adorable, it's a cockroach. Cool, right? So yeah, we do bug programs and insect programs. So we actually bring a cockroach out. And so that is not the reaction I often get. I have parents running away terrified from me. But basically when it comes down to making a call to action, you gotta choose it wisely. You gotta make sure it is effective and gets your point across. So specifically with the marine mammal programs at the Vancouver Aquarium, I know this slide is a bit chaotic, but these are some of the main calls to action that we use. You can contact the Marine Mammal Rescue Center if you see an injured, orphan marine mammal, even a sea turtle. You can choose ocean-wise seafood if you're watching our Strait of Georgia program all about sturgeon and rockfish. Don't eat rockfish, they're not sustainable, but there's other options out there for you. So we really try to give our audience really accessible things that they can do. Whether it's supporting research globally, managing their garbage to prevent entanglement. As you can see there's a California sea lion with entanglement injuries on it. Whether it's doing net studies, or one of my favorite calls to action that I've been using a lot more recently is inform yourself. Just take the time, ask questions. Don't just accept what's being given to you. Think about the world around you and go find your own information and share that information with friends and family, because science is important. Now, I got the email about this being transmitted globally. That is not my cartoon, but no word of a lie. That cartoon comes from Big Fat Whale. So there you go. You can check out their stuff. So what I did was I ended up watching several of our dolphin programs at the aquarium, and we have 2 different types that we do. We have a Caring for Dolphins program, which gives people a better understanding of how and why we train the animals and why they're actually at the aquarium. Our actual dolphin show is more about BCCSN, which you heard about earlier, reporting cetaceans, and it's a really great call to action. So What I did was I actually watched 12 different programs, and I'm still in the process of doing this, but after each program, I was able to talk to people and find out what they had remembered from the program. So the question I asked them was, based on the program you just watched, what can you do to help dolphins? So I found that 29% of the people I spoke to, of 55, did actually regurgitate BCCSN, the whale report app. They knew about it. They remembered it, partly because the interps are awesome and they say it a whole bunch of times. It's really cool. And people would come up and tell us stories about the time they saw this dolphin 5 years ago, and we're like, you can still report it. It totally counts. We also had 20% of the people saying membership. That is a call to action in the Caring for Dolphin program. So they were remembering it. And the really great thing is that these were the two highest numbers I had out of all of my stats. The last one there I put up, 18% were saying, don't litter. We don't mention that in the program. It's kind of funny and it's kind of sweet too. I guess you put people on the spot. What can you do to help dolphins? And they're throwing stuff at you. True, this does help dolphins, even though it's not directly in the program. But it's nice to know that a lot of the people that we do have coming to the aquarium already have that eco-conscious mindset. And we're just trying to foster that and make it a bit bigger. So there you go. Download the app. It is free. Whale Report, iPhone, Android. You can get it. You probably see more whales than I do. Other really cool things I found by talking to our cetaceans research team was they had done a survey themselves of the website users, because you can report not only on the app, as you've heard Jevin say, but also on the website. But 12% of 439 people surveyed heard about BCCSN by coming to the aquarium. That doesn't necessarily guarantee they saw it at a program, but hopefully they were talking to an interpreter, a staff member, a volunteer, so they are hearing it from us. And then 12% of 323 people surveyed had heard about the app from the aquarium as well. And it's also, again, back to the whole point of people are kind of eco-conscious. 83% of the people that were spoken to at an outreach survey that BCCSN did, they just wanted to do something to help protect cetacean and sea turtle populations. So, what does this all mean? It means that, first of all, interpretive programming at places, not only the Vancouver Aquarium, but anywhere, if you have a facility, get interpreters. We are effective. It's a really great way to connect people to the resource that you're trying to share, and it's a really effective way to get people to become active. So that's one of the take-home messages for you. Absolutely, we can be more effective, and that's a goal for myself for this coming year. I want to see if there's a different way to word our messaging, whatever it is. How can we be more effective to reach more people so we can get the story of echolocation studies or porpoise information or whatever it might be? How can we be more effective? Most importantly, we do love to talk, and if you give us enough time, we can pretty much connect anyone to anything, as random and strange as it might seem. So if you want your stuff talked about, come on over, find me. I'd love to talk about your stuff and work with you to be able to share more science, more research, more information about these amazing marine mammals that we are all here to celebrate. So definitely come and find me, and we can chat. Thank you so much.
Questions? So maybe something I'm just curious about is, it's great to see that 83% of the people want to do something. That's great. I wonder about the other 17%.
Find them and beat it into their heads.
But I'm still, but it still seems that when the percentages with the picked up messages is not very high. And is it that they're not listening, or that just have to find better ways to do it? So what's your comment on that?
Yeah, it's one of those things where the numbers aren't as high as we'd like to see them. Historically, we have kept data over the years of our programs and the effectiveness of messages and what people remember. We're trying to get better about managing that and evaluating that at the aquarium. So we've been working with our membership team and our guest services team that are kind of at the front lines. And we're really trying to find ways that we can survey people. So ideally what we want to do is ask people a month after their visit, 2 months after their visit. What do you remember about your visit to the aquarium, and are you still eating sustainable seafood? So ideally, the number would increase in that case, but yeah, the number's still low. There's a lot we can do. I'm up for suggestions too. If you have ideas that you've seen or you want to try, you can always talk to the interps, and we're definitely down for a conversation, but we want to get that number up for sure.
And I guess I just add to that, you know, in the science side, we have the same challenge. Through science, we want to affect change, but is our message being read and put into action? So it's the same challenge we have as well.
Definitely.
Yeah. Okay, thank you, Lauren. Our next speaker is Chad Nordstrom. Unfortunately, Ed fell ill and is unable to come talk about sea otters and kelp, but I'm sure we'll have him here next year. And I know many of you are looking forward to that as well. So Ed, if you're listening, we need you here next year. Chad, please come up.
Yeah, terrific. Thanks so much. Thanks, Andrew. You can definitely count me in as one of those who wants to see what Ed's been doing and what he's up to. But this afternoon, I'm going to give everyone here a brief recap of our recently completed field season, looking at non-estuary seal haulouts for scat samples in an effort to describe diet for harbor seals in the Strait of Georgia. Now, I'm the one up here giving you a brief talk, but nice. But my partner in crime this past 7 months has been Sheena Majewski from Fisheries and Oceans Canada. She's a PI on the project, as is Andrew Treitz here at UBC. And I was made available for the project from my regular position at the Coastal Ocean Research Institute. Now, so what do I mean by non-estuary? What was the rationale behind the project? Well, really, we're looking at and targeting these sort of rocky islets in the Strait of Georgia to complement some of the more recent estuary-focused studies, and that was looking at the predation of Chinook and coho smolts. That was work done by Ben Nelson and Austin Thomas here at the Marine Mammal Research Unit. As a bonus, we were going to be able to update some of the non-estuary diet information for harbor seals in the Strait that was really last focused on in the late 80s and early 90s, and there's some people in this room that contributed greatly to those studies. And ultimately, we wanted to be able to ask, are the diets that have been recently determined from scats collected in estuaries more representative of diets in the Strait as a whole? Now, our project objectives were to collect 70 seal scats per month from April through November this past season. We were looking to also add a geographic component to distribute our sampling effort. As you can see from the survey map that's come up here on the right, harbor seals make extensive use of the Strait, both from the north up near Quadra Island all the way down to the south at Race Rocks. And eventually we're going to undertake a DNA and hard parts analysis. So this is a full disclaimer. There are no results in this talk. But we will tell you about them next year. Really, we're going to sort of sum up some of the challenges and some of the successes that we had this past field season. And ultimately, we want to be able to compare our 2016 diet findings to estuaries from previous years, to non-estuary sites in the 90s, and then in between regions in the Strait this particular season. So what did we accomplish? Well, you guys are lucky. Our field season just ended on Monday, so these summary totals are brand new. We were able to sample 7 non-estuary sites repetitively throughout the summer months and through the spring through fall, and fairly good distribution from north to south through the Strait. We were able to maintain 1 estuary site, that's at the Cowichan Bay log sort, to kind of keep that data series going. And then we were able to expand our geographic range during the summer months, both when the weather was a little bit better, those boxes are outlined in orange, and also by being able to partner with other networks that were available and out there, particularly the Pacific Salmon Foundation Citizen Science Project, and they're out collecting oceanographic information in some of these same areas. And I'd like to call particularly attention to a couple of different sites. We've got Snake Island just outside Departure Bay by Nanaimo, and then a pair of sites down at the bottom of Harrow Strait, and that'll play into some of the challenges that we saw this summer. In terms of numbers, we were able to collect just over 1,100 non-estuary scats and add another 300 from that Cowichan Bay site. We mostly met our 70 scat monthly quota, particularly because those scats were challenging to find, and congratulations to everybody out there who could see those scat samples on the slide without the aid of the red dots. They're really indicative of what's available out there, particularly early in the season. So if anyone's worked on stellar sea lion scat samples, northern fur seal scat samples like I have, they're much more easy to find compared to these ones. And really that's driven primarily by seal behavior, and I'll try to show that in this next figure here. So this plot is of all the non-estuary scat samples that are collected by month. So on the bottom are the x-axis, we've got months, and on the y-axis is total samples. And the very first thing you should notice is that we've had to pool in that first bar, April and May, just to reach 50 samples. This is incredibly important because this is when salmon smolts are running, and it has an incredibly important component of this study as well as for the ecosystem. And it really had nothing to do with the amount of effort that was going in. So the numbers that have popped up there is between 8 and 10 sampling sites per month. And I'd argue strongly that we spent more time per site early on in the season looking for those scat samples without the numbers to show for it, as we did early on later or towards the middle of the season. Particularly those peaks, July through mid-October, which coincides with pupping and molting. So the seals are hauling out earlier, they're hauling out later. Of course, the tides are better, so those samples are being deposited, and they're much easier for collectors to come by and collect them afterwards. Another interesting impact, that's one of those challenges, was the impact of site selection. So this is a shot of Miami Islet in the Northern Gulf Islands. Really good example of a multi-tiered site that can provide samples over a range of conditions. So Sheena there is walking along the reef, which is normally exposed at low tides, as well as sort of this upper tidal area, upper rocky area with rigorous topography, able to sort of trap scats and leave them there for researchers. First bar plot is of the Snake Island site, previously identified as being a very productive site from those past examples, and past work in the 90s. And you can see that it follows the same general trends. Even though there's not a lot of samples, at least it was producing early on in the season, and then it follows and spikes in the middle of summer. You can compare that pretty drastically with some of the lesser known sampling sites in Harrow Strait, where we were doing a lot of hunting around early on in the season, and those numbers are incredibly low. We were working there in the Great Chain Islets and Chatham, out of Oak Bay in the south part of Harrow Strait. Hundreds and hundreds of seals, but the islets there just simply weren't retaining samples for us. So we moved 15 kilometers to the north to some more of these sites that more closely resembled the snake site at Sydney and Darcy Islands, and those numbers jumped up accordingly. So that isn't to say that the Oak Bay sites won't potentially produce, we just haven't managed to crack them in that first. So the take-home message is we were able to collect over 1,000 non-estuary scats over the 8 months of the program. 8 of those sites were pretty regular, and then we expanded them as possible, particularly with the use of that aforementioned Sightings network or oceanographic network. We modified our sampling areas as needed throughout the season. And really, it's those fine-scale site characteristics that have a lot to do with how samples are resilient to seasons, weathers, various tide conditions, and just relying on the number of seals in an area simply isn't enough to guarantee that you're going to have a lot of samples. So looking forward, of course, sample processing is continuing and will continue through the winter, which will provide the first kind of information that we'll be able to get for our genetic and heart parts analysis. And then hopefully, with a little bit of luck, we'll be able to fill in some of the sampling gaps where needed, particularly by expanding that network and those relationships, and working more in the spring and in the shoulder seasons. With that, I'd like to thank Pacific Salmon Foundation for their generous funding of the project, and Andrew for championing the project here as well. To my colleagues at the Coastal Ocean Research Institute for putting up with my highly variable schedule the past 7 months, and to all of the colleagues at Cetacean Research Program who jumped in as volunteer assistants and also as boat drivers, particularly when the weather got lousy. I'd be happy to have any questions. Thanks.
So any questions about Chad's passion?
It is. It's the world's best treasure hunt.
Yeah. I might just give a bit more context to this. So what Chad is doing is a piece of project. It's a $20 million 5-year program between the United States and Canada focused on the Salish Sea. And the primary question is, what's happened to Chinook and coho? And so the studies range from predation, changes in primary productivity, to changes in forage fishes, so is oceanography. It's got not just NGOs involved in collecting data, there's DFO, there's universities, and hopefully, I think we're in year 2 process. By the year 5, we should have some solid results. Whether or not we're going to be able to say why we're missing the fish is another matter, but we will certainly know an awful lot more about the Salish Sea than we do now. We're going to hear from Hassan Alec next, and he's going to tell us a bit more about that harbor seal predation component. Want to come up? very much, Chad. And just before he starts, I might just make a comment that, we used to sort of dismiss seals as having a big role to play in the decline of salmon, but we're really just thinking about the adults. And the work that Hassan is doing and others as well is pointing to maybe we've been looking at the wrong end of the life cycle. The real problem is with the small, early stages. Go ahead.
Thank you. So thank you, Andrew. So yeah, my name is Hassan Aleg. I'm a master's student here at UBC with the Marine Mammal Research Unit. And for my project, I used really fine-scale biologging data to investigate on the variability of harbor seals' foraging behavior during the outmigration of the juvenile salmon in the Salish Sea. So this project, as Andrew mentioned, is a part of the Salish Sea Marine Survivor Project, which aims to restore the habitat and production of coho and Chinook salmon in the Salish Sea. So the Salish Sea is the largest inland marine ecosystem on the Pacific coast of Canada and the US, and historically it used to have abundant and sustainable Pacific salmon populations. But since the 17th, some of these populations, like coho and Chinook salmon, have drastically declined and were never able to recover, despite all the fish recovery efforts that have been initiated. So one of the main hypotheses to explain this recovery failure is the increase of harbor seal predation pressure. And some recent studies show that there is increasing evidence that the predation of the seals on the smolts, which is the juvenile stage of the salmon, may contribute to the poor marine survival of Chinook and coho salmon. So far, all the studies have focused on the diet of the seals. This is what Shad explained in the previous presentation. But really, there is really few information about the spatiotemporal variability of their foraging behavior during these migrations. So the question that I'm addressing in this project is to try to understand how, when, and where the seals are eating these smolts. So to do so, we equipped 17 seals in the Big Walicum area, which is on the east side. I don't know, we don't see the pointer here, but it's on the east side of Vancouver Island, over here. And so we equipped 17 seals with biologgers that collected fine-scale data on their movements and behaviors for 1 month in May 2015. And during that time, there was 350,000 coho and 3 million Chinook were released from that area, from the Big Walicum hatchery. So here we have all the GPS locations of all the 17 seals that we equipped, and we can see that the majority of the seals stayed within the Big Walicum area, where they conducted really short foraging trips around their main haul-out sites. But 20% of the seals traveled long distances towards Parksville, over here, or Malaspina Strait, Howe Sound, and even Indian Arm, and they used different areas in the Strait of Georgia. So since I'm interested in the interaction between seals and outmigrating smolts from the Big Walicum hatchery, I focused my analysis inside the Big Walicum area. And based on the feeding intensity of the seals inside the estuary where the smolts leave the river, I found 3 different foraging strategies. So the first one has seals that don't really respond to the smolt outmigrations. So as we can see in this map, there is the feeding density of the seals, where green is low and red is high feeding density areas. And we can see that the seals fed all around Hornby Island and mainly around Norris Rocks, which is the main harbor seal haul-out site in that area. And using this figure over here showing the vertical distribution of the feeding event, where the surface is at 0 depth, we can see that feeding was happening from the surface to 200-meter depth, which suggests coastal, pelagic, and benthic feeding. So for the second group, we have seals that fed mainly over here between, in the open waters between Norris Rocks and Big Qualicum Estuary, between 100 and 200 meters, and they also occasionally fed inside the estuary. For the last strategy, the seals fed around Flora Island over here, between 100 and 200 meters, but these seals were specialized on feeding on outmigrating smolts in the estuary, where they focus all the feeding intensity inside the estuary. So if we stay with these small specialist seals, we can look at their feeding intensity inside the estuary for each day of the study, and we can see that there is a clear increase in their feeding intensity just after the release of the coho salmons. And that matches exactly the number of smolts that were released from the hatchery. But we don't really see the same increase in their feeding intensity during the outmigration of the Chinook. And that could be due to the fact that Chinook smolts are 3 to 5 times smaller than the coho smolts when they leave the hatchery, and they're more likely to be below the size threshold of 10 centimeters, which is here, which has been proposed to be the minimum size of harbor seals prey. So what the seals are eating during that period? So the hypothesis that I think is the most likely, for me at least, is that the seals target the predators of the smolts and not really the smolts themselves. And that's it. So just to sum up everything, from this study, I found that the estuary, the Big Qualicum Estuary, was a foraging hotspot for these populations of seals. And although 50% of the seals used the estuary, the estuary was an important foraging area only for 80% of those seals, which is 3 seals of 17 seals that we equipped. And the small specialist seals seems to take advantage of the coho release and not really of the Chinook release. Thank you very much.
Do you have any questions?
Yes.
Thanks, Hassan. That was great. What was your metric for feeding intensity? How are you quantifying that?
So yeah, I didn't explain that because I didn't have a lot of time to explain the method. But basically, I used the seal body acceleration as a proxy of feeding. So basically, when the seals they do really intense acceleration, so I assume that's like the seal is chasing or capturing a prey. Of course, it could be other behaviors, but I think the majority of those behaviors are probably feeding. Yeah.
Hassan, could you also comment on the pit-tagged fish that were done in terms of knowing how you related that acceleration signal to consuming coho?
What do you mean exactly?
Well, you just maybe just mention that.
Okay.
The head-mounted tag, so we have a bit more information about this specializing on Chinook.
Yeah, I didn't talk about this part of the project because it's not from my thesis, but we also equipped the seals over here with a pit tag reader on the head, and basically the hatchery tagged I think 35,000 of smolts with those really small pit tags, and every time each of these seals eat one of those pit-tagged fish, we can detect that event. So that study was done by Dr. Austin Thomas, and according to his all his calculation, they were like eating, I think, around 6% of the release of the coho, and not really surprising. all the small specialist seals that I found are almost all the seals that they had like pit tag detections during that period of time.
Another question?
I was wondering if you know if are there any hard parts from these very small smolts that would end up in a scat?
I would say yes, but what Austin has developed is a method using DNA to quantify the number of the species and the quantity, the proportion of each species in the diet. Yeah, there is probably a problem when the size of the fish is too small. So there is a problem that you might not find any trace of bones or otoliths in the scats. But I don't really know more than that.
And this is where Chad and Sheena's studies coming in, using both DNA and heart parts, but also looking at the timing of when those fish show up in terms of the months, because certain adult sizes should not be present at that time. Any other questions? Oh, way at the back. I'm not sure if I heard the question either. So the question is whether or not wild Chinook salmon are below the 10 centimeter size, given that most of the study here is focused on hatchery.
So in that river, there is wild Chinook salmon. I don't know, we don't really know exactly the timing of the migration, but it's between April and June, somewhere there. And I tried to ask the hatchery about more information, but they don't really know like the average size of the wild Chinook. So we know the ones coming from the hatchery because they have to measure them. But from the wild population, I don't really have that information. Yeah.
Can I just ask, are the smolt being released and the Chinooks, are they being released at daytime or nighttime? Did they give you that data, and does it make a difference?
Doesn't really make a big difference because I think how they do that, they just open the door and they just let them going. And the smolts are known to migrate during the night. So they're not really active during daylight. And I didn't show these results here, but we can see that the highest feeding intensity in the estuary was during the dusk, at the end of the day and the beginning of the night. So this kind of makes sense because the smolts are more active during the night. So the seals have like really good capacity to detect prey in a really low light conditions. So that was like the optimal time for them. At least it's easier time for them to catch the smolts. So I don't think the time when they release the smolts at the hatchery will change anything because the smolts decide when they start migrating or not.
I think we'll cut it there. So thanks very much.
Thank you.
Oh, and I might also add that the seals are just one on a list of predators. Everybody loves salmon. And discovering that in some of the rivers, raccoons are major predators, mergansers, and also garter snakes. And they're finding in some of the fish traps, monitoring the movement, they're finding them full of snakes that are also eating fish. So we're learning so much more about how this whole system works in such fine detail and really changing our perceptions about the ecosystem. Next up, we have Aaron Purdy.
All right. Hi, everyone. Yeah, as I said, my name is Aaron Purdy. I'm another one of the Marine Mammal Research Unit's master's students. And for this past year, I've been studying diving physiology in stellar sea lions. This is one of those stellar sea lions right there by the name of Sitka. So for this presentation, I hope to give you just a brief overview of what I've been doing over the past summer and some of the goals that I hope to accomplish by the end of my master's. I also hope to convince my supervisor that I haven't been completely wasting his and everybody else's time. We'll see if I can do that. So first off, a little overview of why studying stellar sea lions is important. So as I'm sure many of you know, from the late 1970s to the late 1990s, there's been a huge population decline in particularly the eastern population of stellar sea lions. Along the Gulf of Alaska in the Aleutian Islands, kind of where at the bottom of those red dots right there, we saw declines of more than 80% in some areas. So trying to figure out why some of these declines are happening. One possibility that might be causing this is that there's been a shift in prey. So it might be that these animals are no longer able to meet their daily energetic requirements. And because of that, it's really important that we understand their foraging capabilities. So understanding how well these animals are able to dive, so we know what happens when they're pushed to their limits, which might be happening in the wild. So one of these limits is called the aerobic dive limit. In the diving physiologist world, this is, people are kind of obsessed with it, but it is for good reason. The aerobic dive limit is defined as the point in a dive in which you begin to accumulate lactic acid in your system. And the reason you do this is you're switching from aerobic metabolism to anaerobic metabolism. It means you're using up your oxygen stores, you're reaching the ends of your limits right there. Diving past this point is associated with different costs, and you can kind of break these costs into two different groups. First is an energetic cost, so it takes energy to rid the body of these metabolites that are building up in your system. And the second is a time cost. So the only time in which you're able to actually rid your body of these metabolites is while you're at the surface breathing oxygen. If you're at the surface, that means you are not foraging. So if with an increased recovery time, you're going to have less time to eat, essentially. So what I'm interested in is looking at how recovery time is related to this aerobic dive limit. So this is something that I kind of expected, one of my predictions when I was going out with beginning my research, that with an increase in dive time, you're going to have an increase in recovery time. That just makes sense. The harder you work, the more time you have to take to recover. But I was expecting once you surpass that aerobic dive limit, you're then going to have a disproportionate increase in recovery time because you have to spend that much more time recovering from all of these metabolites accumulating in your system. So my research goals, I had two main things I wanted to set out to do. Was one, determine the aerobic dive limit in stellar sea lions. This hasn't been done in very many pinnipeds at all, and so this is something that'd be really interesting to see. And then also looking at how recovery time changes with increased dive effort. Another way of putting that is seeing how recovery time changes after they exceed that aerobic dive limit. So how do you do that in a bunch of sea lions? I've been lucky enough to work with, in partnership with the Vancouver Aquarium at the Open Water Research Station. Out there they have 4 trained stellar sea lions. You saw one at the beginning, that was Sitka. This one here is Bonnie. You can tell by those beautiful long whiskers. She's adorable. These animals are incredibly well trained. They're actually able of swimming, they're trained to swim out in the open ocean and perform dives out there. And so we're actually able to take these animals into a more natural environment to see how these animals are going to be functioning in the wild. On the right, you can see a picture of the setup that we have out there. We have a floating barge where we do most of our data collection, and I'll give you an overview of how that works. So first off, I'll show you kind of the setup that we had for measuring lactic acid. We have this floating barge, you can see in those gray boxes there, with fish pumps these tubes that extend down up to 40 meters into the water, so down to relevant foraging depths that you'd see sea lions using in the wild. And like the name implies, fish pumps, we can pump fish down through them. So this encourages the sea lions to dive and then stay in that location. So sea lions dive, they're swimming back and forth between these two tubes, and they're feeding on the fish. It mimics a foraging bout. When the fish stops, they don't see no point in staying there anymore, so they surface, and the trainers then have them come up onto the barge, where we take a voluntary blood sample, and then we use just a small handheld lactate meter, similar to what some people, if you've ever had someone who's training for running, they use something similar to this. They get a small blood sample, and we can measure the lactic acid in their blood. So this is what it actually looks like when you're out on the water doing this. And right now, this looks kind of like a calm day. But if you can imagine, you're out on a floating dock. It's rocking back and forth. There's wind, there's rain, there's boats driving past. It's incredible that these trainers are able to get this blood from these animals, particularly because they're completely wide awake the entire time. They are voluntarily giving that blood sample. It just shows how great this connection these trainers have with these animals, and just how much work they've been able to put into it. It's really fantastic. So then that is the how we measured lactic acid. Another part of the study was measuring that recovery time. So similar setup where the sea lions dive down, they're feeding on fish, but when the fish stops, they then, instead of surfacing and coming up onto the barge, they instead surfaced underneath this metabolic chamber. It's a sealed chamber where we have air flowing over them. They're breathing in oxygen, they're breathing out CO2, and we can get measurements of both of those gases and use that to measure their metabolic rate. So after a dive, their metabolic rate is going to be increased. We're measuring how long it takes for them to get back to that normal level, determine how long it takes for them to recover from that dive. So things that I still hope to do, I'm going to analyze that recovery time for oxygen and CO2 and see how they interact with that aerobic dive limit. Then hopefully I'm going to be able to use all of these together to model different foraging strategies that these animals might be using in the wild. So hopefully I've been able to convince you that it hasn't all been fun and games out there. I have been doing some work despite what looks there might be. A big thank you to all of the staff at Open Water and to all my other support. If you have any questions, please ask. Thank you.
Do you have any questions?
The main question, I assume, is working with sea lions that cool? And yeah, it is.
Yeah, we'll let you off the hook with that. Thanks, Aaron. And Rhea Storland.
Hello, thank you for the introduction, Andrew. My name is Rhea Storland. I'm a brand new master's student here at UBC in the Marine Mammal Research Unit. And so my project is still really in its infancy stages, but I'd like to give you a brief overview of what I'm interested in studying. So what I am going to be looking at is cardiac function in stellar sea lions. So it was great to have Aaron just speak for me because he gave you guys a really good introduction into stellar sea lions and some of the issues that they're facing. But you're probably wondering right now, why would you be interested in studying cardiac function in stellar sea lions? And the main focus is that there's just a huge gap in the knowledge right now. So with stellar sea lions being deep divers, one of the things that we know about with their dive response is that their heart rate decreases while they're diving. And so a lot of research with marine mammals in general has focused on heart rate, but we haven't been able to look so much at the heart itself. A little bit of that is due to a lack of technology, so we haven't been able to. And I think you can imagine if you tried to attach electrodes onto a stellar sea lion in the wild, you'd have quite a bit of trouble. And so we haven't been able to do that. And so my hope is with the animals at the Vancouver Aquarium that we would be able to look at sort of their ECGs, so looking at the electrical impulses that control the heart, as well as the stroke volume. So that's the volume of blood that's pumped out with each heartbeat. And so basically, since there's just this huge gap in the knowledge, we want to determine what the baseline is. So what does their ECG look like? What is their cardiac output? And I think all of you guys sitting here, you probably know that your heart rate is probably between, 60 and 80 beats per minute, depending on your level of physical activity, your health, and the amount of caffeine that you've had today. Mine up here in front of you maybe is going a little bit faster, maybe closer to 100 or 110 beats per minute. But as you know, just at rest, our heart rate is lower. When we exercise, our heart rate increases. And for marine mammals and stellar sea lions, they have the exact opposite. So at rest, that's when their heart rate would actually be high. And then when they're actively swimming and diving, their heart rate is going to decrease. And so we know that, but we don't know exactly what's going on with the heart. And one of the inherent problems in sort of determining what normal is that there's variation between individuals. So we need to look at multiple individuals to get that baseline reading of what normal is. Along with that, I think everyone can probably agree that there's going to be a difference in heart function between terrestrial mammals, like the dog, as opposed to marine mammals, but we don't know what those differences are right now. As well, we might find that just between different marine mammals, there's going to be differences in heart function. So we know that phocids, so the true seals, they are better divers than the otariids, so your fur seals and the sea lions. And so potentially there could be something inherent in their heart function that might sort of connect to their dive capacity. And it would be really interesting to sort of be able to see that. And so what kind of tools do we have in order to look at heart function? On the left there, we've got electrocardiography. So this one you guys are probably familiar with. You've seen a TV show in an ER, or possibly you've had an ECG done on yourself. One of the things, though, with the ECG is, in general, we think of it for thinking about heart rate. But all of those different inflection points, their amplitude, the distance between the inflection points, those are all important things to look at as well. And so that's where my research might focus in on more. And then over on your right, we've got electrocardiography. And so that's where you can actually get an image of the heart, and you can look at that volume of blood that's being pumped out of it. So, as I mentioned, very much a project in its infancy stages. So please, if you have any suggestions or if you know of any literature, let me know, because I've had a lot of trouble even finding a single baseline ECG for a stellar sea lion or another pinniped. So please come find me if you've got any suggestions.
Do you have any questions? So I know there's at least one vet here who spoke to me. You need to meet Rhea, because some of the knowledge you have in doing this sort of work on animals is probably transferable. Any idea, Rhea, why there seems to have been so little work on heart rates, given that we know they've such incredible control over heart rates?
Yeah, so heart rate, sort of understand, and we understand how it functions during a dive, but thinking about cardiac function in terms of stroke volume hasn't really been looked at, again, because of the technology. So doing something like an ECG, or not an ECG, the echocardiograph would be very challenging to do. So it's just not something that I think has been able to be done yet.
Right, thanks very much. Now, a scientist who needs no introduction, David Rosen.
Afternoon. For those who I haven't met, my name is Dave Rosen. I primarily, during the day, run the captive pinniped research program, both at the Vancouver Aquarium and at the Open Water Research Site that we've heard a little bit about already. I'm starting my talk sort of uncharacteristically with a very serious quote, because, you know, I always give a serious talk. And also, I think it kind of sums up my interest as, quote, a physiologist, although I hate that term, of my interest is trying to, in the conservation view, try to figure out what the connection is between changing the environment and changing the population. And I view that as what is happening to the individual animal. So how does physiology and behavior, how are they affected? How is that ultimately going to drive population? Because that's the individual animals, the link between change that we're all observing on the oceans and the change in the numbers that we're all worried about ultimately. So I've been doing this for a while. So I wanted you just to cast your minds back to 2003. And 2003 was kind of a benchmark year for a lot of new stuff and experimentation and science and living on the edge. And so we had things like the completion of the Human Genome Project. We had Galileo crashing into Jupiter on purpose. We elected, in the States, a totally unsuitable candidate for political office. Never going to see that happen again. And probably the biggest experimentation of the year occurred at the MTV Music Awards. But 2003 was also notable for the opening of our open water research station in Port Moody, which Aaron gave you a brief overview of. It was, the grand experiment for us. We really had no clue whether it was going to work at all. We started small with an animal, and over the last, it's been 13 years, 10 years since we've actually been publishing hard data on this, and so I was asked by an editor of a journal, he said, "You've been talking about this for a long time, Dave. Can you put together a synopsis of what you found, what your scientific journey has been with this rather remarkable scientific paradigm that we've established," which is really there, again, to emphasize, before that, and we still have animals at the Vancouver Aquarium, who's one of our valuable partners in this project, but we are always physically limited at the aquarium by pool size. Marine mammals are really good at diving and swimming, and they can only do so much of that, even in your biggest tank. And so 13 years ago, Andrew Treit said, let's take them out and see what happens. Let's take them out where environmental constraints are not an issue. And from then on, we found some really amazing things. And so I got to sit down, and over about 2 years, I got to write up what I've been doing as part of a vast team at the Open Water Research Site. And this just appeared this year online. It'll come out in paper copy if they still have those things. And it's a summary of our scientific journey with the animals at Open Water Research, just focusing on the diving physiology side of things. So we've done a lot of other cool stuff as well. But that's what this article was about. And so I thought what I'd do is give you a really, really brief overview of some of the things we found over that years. One of the things that was fun about this, no, this isn't the overview. This actually appears in the paper. And one of the reviewers, we nag reviewers because we think they just make life hard, but they actually suggested do a flow chart. And we found out this flow chart not only highlighted the questions, which are kind of in the grayer boxes that our central experimental questions, but also the flow of science, of what one study led to understanding one thing, which of course led to more questions, which of course is what science is all about and why it's exciting. So I'm going to take you through some of the highlights of this. Hey, download the paper illegally. That'd be great. And take a look at some of the details. And so what I thought I'd do is I'd take it sort of, what's the approach? What's one of the audiences for this type of stuff? And that is scientists trying to talk to people who are invested in the answers. So I've got, Mr. Typical Scientist there. Some similarity there. Certainly, this part. And who's he explain it to? And usually we have to explain it to resource managers. And because we're in the conservation business, that's the action point, is getting it to people who are making the management decisions. So I needed something that properly demonstrated management and government workers. So I have Patrick here. Graham, I know you're not offended because you're retired now from DFO. So this is sort of my short explanation of a conversation between these two of what we found so far. And so the most obvious question, and the one we started with in our program, and we naively thought this was going to be this most thing, is what is their metabolic rate? What is the cost of diving? And we thought this would be the easiest thing. Because as Aaron talked about, and as Ria alluded to, marine mammals are really good at down-regulating everything when they're diving. And so the general impression is it doesn't cost them anything to dive. In fact, it's probably cheaper for them to dive than to not dive. And we did find that the cost of diving could be really low for these guys. But they weren't really diving. They were basically cowering at the bottom, not moving. Okay, so yes, they could batten down the hatches and have a really low cost of diving, but that's not what they're normally doing. So if that's not what they're normally doing, what is the cost when they're normally doing? Do we have a cost? Well, sort of, but it depends. Depends on what they're doing, what kind of dives they're taking. Is it a series of dives or a single dive? How deep are they diving? How long are they diving? What are they doing underwater? There is no single number. Now, is that because of this great dive reflex? So in other words, if it's not costing a lot, is that because their heart rate's dropping? Things like that. Again, sort of. Yeah, we could map out all these great physiological adjustments that they were making, and that allowed them to stay under. But unfortunately, that didn't always correlate to how much energy they were using during the dive. And that becomes important because we wanted to know, could we use measurements like heart rate or activity to predict how much energy they're using? It's one thing to measure these Steller sea lions out open water, but we want to measure the energy ultimately of sea lions out in the wild. So can we just use things like heart rate or body movement through accelerometers to, here's the Fitbit. Can we attach a Fitbit to these guys or a heart rate monitor and predict how much energy they're using? Well, sort of. Again, under very gross circumstances, these type of measures work, but we were kind of the dragon slayer in a lot of these. When we looked at fine scale changes, it turned out a lot of the technologies, which in some cases scientists were already using to study wild Steller sea lions, weren't as strong as they appeared to be under some laboratory conditions. The other thing we looked at, again, Aaron already sort of alluded to this, is we wanted to know what the reactions were of these sea lions and what the costs and benefits of changes behavior were when they were faced with different foraging conditions, whether it was deeper prey or more abundant prey. So one of our studies, several of our studies actually looked at what happens when you change prey abundance. How do the sea lions react? Is it better for the sea lions to have more prey, or does it really not affect their behavior? And we found that, yeah, not surprising, it was better for the sea lions to have more prey at depth. And not surprising, when there was more prey at depth, they stayed down longer. It was a good spot to be. Problem is, then they had to recover longer. Aaron already talked about, you stay down longer, you have to recover longer. So that was the trade-off they had to make. But then when we modeled that behavior, we found that overall, they actually gained more energy. And that sounds great. Got more food, you gain more energy, that's fantastic. Okay, let's reverse that though. If food is more scarce, proportionally, you're getting even less energy than you would predict just by counting how many fish there are. Now, this is the question at this point the managers are usually asking the scientists. Here's the answer we'd all like to give. But scientifically, the world is complicated. Animals are complicated. Physiology is complicated. The interaction between the environment, the physiology, and behavior of the individual and populations is really complicated. People are always going to be pressed for simple answers, but they're not necessarily going to be more accurate or useful answers. And so I think one thing that our study has done is try to describe in a simplistic manner without oversimplifying how changes in the environment could be affecting individual animals without giving a single number, without giving a single answer, to show the complexity in the system that you need to understand in order for your predictions to actually mean anything. The one thing I'd like to do is usually at the end of the talks we have a big figure about all our funding agencies, and I don't think that's the important thing with something like the Open Water Research Station. I think what's important is it takes a village, literally. Perhaps a small town. We have the researchers, the grad students, who are all working hard out there. We have the animals, of course. We have our employees and our partners at the Vancouver Aquarium, the trainers, the vet staff. This is a nice day. Most of you live on the West Coast. You know what it's like 8 months of the year. Imagine working out. And so co-authors get recognition on these publications, and what doesn't get enough recognition is the tremendous effort of all the people who have supported us over the last 13 years on doing what we think are really good scientific experiments at the Open Water Research Site. Thank you.
So, pretty complicated. Anybody have any questions? I'm just wondering how...
Sorry, could I just say one thing? I wasn't joking about you can download, just don't tell anyone. The publication, I believe, is up on our website. So if you want to, don't tell anyone. Just between us and the 1,000 people online. Just between us. So I'd encourage you, and it's written sort of like more like a story than a normal scientific journal article. So
Dave, still when we get back to the managers,
yeah,
they're concerned that sea lions can't get enough fish.
Right.
And so how do you take your physiology to help them understand the ability of sea lions to find fish, or if they can't get enough fish? How do we make those links?
Well, that's kind of the whole endeavor of the project, is to design experiments under controlled conditions, which are artificial, and apply them to the complicated world that's happening, in the wild. And so you have to do it through clever design and careful interpretation of the results. And what we do is we say, okay, what do you think is happening, as managers or other scientists think is happening in the wild? Analyze one aspect of that giant change that is happening and saying, okay, let's manipulate that in a controlled environment, just like you do with most science, and see if the results with the individual sea lions, whether it's physiology or behavior, changes in the direction we would suppose. And does that lend support to the hypothesis of what's happening with animals in the wild? So it's a supporting or a non-supporting documentation of the facts through careful, controlled experiments. But we're not trying to, nor would we ever try to duplicate what is happening in the wild. And sometimes they have trouble with that. Managers go, well, it's not wild sea lions, therefore, it's not the same thing. Well, thank God it's not the same thing, because we can actually interpret results.
Dave, would it be too simplistic to think of it this way? From the point of view of a fish, the water doesn't contain much oxygen, so a marine mammal is like a rocket ship coming down from above, packing all this oxygen from the surface, and is capable of doing things that are energetic, so therefore having a high metabolic rate. So it's not much use. So something like sea lion should have that ability to be highly energetic down below. And take advantage of that asymmetry. On the other hand, crap happens, and if a sea lion goes, or a diving animal goes down, it may not be able to get up again too quickly. A predator, one of my animals, swims along and confines it to a crack on the bottom or something. So, I mean, is that partly? Do you think? Is that thinking wrong? And would that be partly why there's this sort of complex range of results in your study?
I think that gets back to it, and that's historically when we study seals, and they said, oh, there's great divers. They took a seal and they stuck its head in a bathtub, and they went, wow, it can hold its breath for a long time. And they thought that was the diving response. Well, it's similar to diving response, but really it's the fear response. One of the huge advantages of having really well-trained animals is we can separate out not just the fear, but the way we conduct the trials. It depends on whether the animals have control of the situation or don't have control of the situation. That can tell us whether it's a usual or a maximum response. So in other words, a case like that about the killer whale predation would be the same thing. If we have a sea lion go down and it decides to quit after 2 minutes, that is different than having a sea lion go down. It doesn't know how long it's going to be down, and we allow it to quit after 2 minutes. Because the sea lions are going to go, I don't know how long I'm going to be down. I'm preparing for the worst, under the second case. Which brings up another debate that's just starting to come in, this clarified in the cetacean research, is when we talk about the dive reflex, physiologists traditionally have thought about this as an automatic response. In other words, you can't have, outside of the fear response, the animal has no control. It's, oh, I've been down here for 3 minutes, so therefore I'm doing this. I'm down here for 4 minutes, I'm doing this. I don't prepare for dives. Well, now we've seen just recently in the cetacean literature that in fact, not surprisingly, they do have conscious control of things like heart rate. So, I mean, my belief has always been that that animals do have control of their physiological response within a degree, and therefore you have to be careful how you're measuring the animals to make sure that whether you're measuring a voluntary thing or a maximum thing or a fear thing as well is very important. And I think that's one of the reasons we were getting very low responses at first, is not only were they not moving at depth, but they didn't know how long they were going to be at depth.
And we'll cut it right there. Thanks, Steve. So I think as a number of you have heard, a number of stories done that the future of our open water research station is in doubt, that we've been funded for a long time by NOAA from the United States. And this year the grant didn't come through. Larry Pinn wrote an article in the Vancouver Sun, and next thing you know, I seem to be doing stories every week as we are now reaching out to the community. So I wanted to give you a little bit of the backstory to why this open water site is so important. David has brought so much of that forward already. But I also want to talk to you about one of our sponsors. And what goes better with a biologist than a beer? And so I want to talk to you about North Coast Brewing, who is brewing a beer to support us. You'll see the backstory here in just a minute. So in British Columbia, our sea lions are booming. We're still in this exponential rise, and it looks as though there's no end in sight. They will, of course, at some point hit K hard. We don't know when that's going to be, but we certainly can't forecast it right now. This is a picture taken up at Warrior Rocks, well named, near Prince Rupert. And this is a site that used to be only for haulouts, so non-breeding animals, but pups are being born there now. We're seeing expansions as new breeding sites are being established. We have now the distinction here at Triangle Island at the north end of Vancouver Island to have now the world's largest breeding site of stellar sea lions. That goes from California all the way over to Russia and northern Japan. It's an amazing place for those that have been privileged to go there, and for others, put it on your bucket list. Well, we're seeing. But of course, as you heard from a couple of the speakers, sea lions in Alaska are on the endangered species list, at least the western population, from going through the Gulf of Alaska and Aleutian Islands over to Russia. And it was first picked up, there was trouble in the late 80s, going to the 90s, and that's when I got involved with stellar sea lion research. Before that, I've been working mostly in northern fur seals. And I thought this was a pretty open and shut case. And the first thing I did when I was asked to do an independent analysis is I got access to all the fishing data, because the one thing that was very clear is that the amount of fish being caught and the amount of fishing activity had risen at the same time as sea lions had declined. The other key thing is that the fisheries were focused on walleye pollock, which is the world's biggest fishery, although at times the anchoveta fishery becomes the world's biggest, but it's huge. That's also currently the big prey item in the diet of sea lions. So some people think you don't even need to do science. This is so obvious. But once I got into all the data, realized that there were no correlations that made any sense. We cannot find outside of that gross general picture, but once you get down to the specifics about where boats fished, how much was moved by this area and that area, it doesn't add up. And so that got us going deeper into what the conditions were. What I realized was that we could do certain studies with models and with existing data. We could do certain studies in the wild in terms of tracking animals and doing behavioral observations. But there was a whole set of studies that needed to be done with animals under controlled conditions. We needed to be able to test different hypotheses. We needed to be able to develop some new technologies to validate technologies being used in the wild, as well as explore some of the limitations to answer questions such as, how much food does a stellar sea lion need? If you're going to say they don't get enough, well, let's start by figuring out how much do they need in the first place. You can only do that by getting controlled measurements. So I approached the Vancouver Aquarium in 1992, which may predate some people in this room, and they were open to me bringing some sea lions in. I told them at the time I'd be there for 1 year, and then I'd go. And our plan was actually to go to Bamfield, to the marine station, and build some open water pens there. But there's certain advantages to being in a place like Vancouver in terms of its proximity to the university, the expertise we had of the animal healthcare team at the aquarium, the training support. And so we started there, and we answered a huge number of questions. We're still answering lots of questions. We expanded it to include northern fur seals and brought pups in from the Pribilof Islands and the Bering Sea, because they too have crashed. and we're testing whether or not some of the same factors are responsible. But there were still some limitations. Aquariums, by the nature of the design, have hard bottoms, and they're shallow pools. We really needed to test the real diving abilities, looking at questions such as the aerobic dive limit. You can't do that in an aquarium. You can't do that with wild animals either. So that's where I got the idea, well, let's try to go in between and propose building the open water research station. So in the first place, when I first proposed bringing stellar sea lions in, people said that was the craziest thing. These were monsters. They'd end up breaking down all the walls, and they'd pick up little children and throw them around. And in fact, it turned out that, well, they did have one bad experience years and years previous. The reality is that very few facilities in the world have ever tried to train stellar sea lions. They're the world's biggest sea lion, and they're simply just not well known. What we now know is that they are very trainable, and we have the most amazingly trained animals that can just do. When we said, let's go to the wild, they said, that's crazy, they're all going to swim away. And what was crazier is that when we first took them out, we had them on leashes. And of course we know that's crazy, because if they wanted to, they could have swum with you and taken you back to Alaska. And the reality is they can leave at any time. And sometimes they go AWOL, but either they swim back on their own or they get on board and come back. But they've been very, very dependent. Essentially, it's like if you're out in the woods with your dog and it's not on a leash, there's a trust, and the animals come back. So we've done also some amazing things that no one has ever done in the world. Anyway, we did know that our funding, you depend on a single source because we have to pay for feeding the animals. Anybody who knows things about keeping vessels on salt water or other things, it's constant maintenance. And we also have to pay people. We've got technicians and trainers we have to pay as well. So our annual cost has been about $200,000 to run the facility, and most of that are people costs. And so we started to look around at possible sources of funding, and Brandon Russell, who I'm showing here on the left, was very creative in lots of ways. Not only did he build this restraining cage with PVC piping over here, he also likes beer, and he noticed that this one called Red Seal Ale, and I'm sure this audience can also recognize, is that a seal? So Brandon, God bless his heart, picked up the phone and he called North Coast Brewing to point out their error. And the person who took his call was the owner and head brewer of North Coast Brewing. And Brandon is always one to speak his mind. But Mark Rudrick, who took the call, saw something in this and related to him. And in the end, opened up a huge conversation. We didn't convince Mark to change the label, but it did make Mark consider going one step further and producing a beer initially as a one-time thing to support us, but later on he thought more of that and decided to do one beer dedicated entirely to us. So these are 3 of the beers they produce. The one on the far right is called Brother Thelonious. I realize I'm now selling beer, but one of the co-owners in the brewery was a former DJ who loves jazz music. And so they end up producing this beer where the proceeds go to the Monk School of Jazz. And I understand that they recently, with their contributions, just exceeded $1 million in donations from this. So Mark decided in the end that he wanted to create a beer for us. And so it's called Stellar IPA. It is available in a few stores, but we've been waiting on UBC to sign the legal paperwork to make all this happen. The bottom label, which you can't quite read, says, in support of marine mammal research. And what I want to do is show you a beer infomercial that tells you a little bit of the backstory. It's a 3-minute clip. It's going to take, when David Rosen said that the open water site is like kind of miserable, wait till you see it here. It's almost like poetry on the water out there. So we're going to set that up, have it run here.
And behind the black curtain here.
Because it's too slow to run it within the PowerPoint. And get the lights. changes happening in the ocean. They're getting warmer, they're becoming more acidic, and in some spots less oxygen, they're becoming hypoxic. These are all really dramatic changes, and it's very possible that what we see right now may be the future. The sea lions, they're one of the best species we can be studying to really understand how the oceans work and how they're changing, and also they can help us figure out how we're going to adapt to those changes. There are 5 different species of sea lions. The biggest of them all is the stellar sea lion. A male stellar sea lion can weigh over 2,000 pounds. At the Open Water Research Station, we are testing hypotheses to explain why some populations are increasing while others are declining. Since the late 1970s, we've seen over an 80% decline in stellar sea lions. And we brought trained animals, and we've set up experiments so we can essentially control the environment. We can take them up into some really deep water and allow the sea lions to move in the ways that they do naturally. By working with them, we could get some insights into the lives and the decisions that sea lions make that we can use to interpret the data we're getting from Alaska, from British Columbia, and elsewhere around the world. Each year, we don't know if this is going to be our last year to keep this going or not. We do it on a shoestring budget. And as a result, our staff have also been looking, along with me, for other possibilities for money. One of our technicians noticed red seal ale, and he put in a call. And the person that he spoke to was the head brewer for North Coast Brewing.
I was always interested in the water and always interested in life in the sea, and got my degree in zoology with the thought that I would one day work as a marine biologist. When we ended up here on the Mendocino coast, we started a brewery instead, and so it seems natural for us to support this kind of work. Eventually, we came up with the idea for North Coast Stellar IPA and put together the group that we plan to support, including Andrew at the University of British Columbia, the Marine Mammal Center in Sausalito, and the Noyos Center for Marine Science right here in Fort Bragg.
I was at my desk one morning and received a call from Mark. He had been put in touch with us by virtue of relationships we have with the University of British Columbia. And what Mark was intent on doing was leveraging their work at the brewery with the work of many of us in the marine mammal research community on the West Coast.
Stellar IPA it's a big hoppy beer, like you would expect an IPA to taste like. We use Chinook hops, which are a classic microbrew hop, and it's a very pungent, resinous flavor and aroma. We have our own hopping method that we use to dry hop the beer. It provides a nice aromatic component to the beer that you can get it in your nose really strong when you're sniffing the glass.
The beer is being distributed throughout our entire network in 22-ounce bottles and in kegs, so you can find it in restaurants and bars and hopefully in every street corner market.
We are in the classrooms teaching kids about marine mammals and their oceans. We're out in the tide pools doing surveys to see how our oceans are changing. These are the kind of things that our partnership with the brewery are going to help us continue to grow and build.
This support from the North Coast Brewery allows us to make sure we have all the resources in place to provide the very best care for these animals while learning about their ocean home.
The relationship that we built with these animals is allowing people to get to know them in ways that we've never known sea lions before. And what the sea lions have been teaching us has expanded our knowledge about the entire North Pacific Ocean, and that's helping to conserve them in the wild.
Our hope is that this really helps to raise awareness for what is a very serious environmental problem.
So 2 things to take away from this. One, if you're a marine biologist and you can't get a job, make beer. Because ultimately you can live your boyhood dream once again, which I think what Mark is doing. 50% of all the proceeds from this come to us to support this program. 25% is shared with the Marine Mammal Center and 25% with Fort Bragg. So it's just a wonderful thing. And it's nice to see when people get that stage in their careers that they can give something back. But we're not out of the woods yet. That's going to contribute a piece We're still looking for more partners. Recently, some of you may have heard that we became a major film site for a new TV series called The Deep. It's all about a marine biologist. They're all very real, except for the part about the mermaids. Just a detail, just a detail. So it's a pilot financed by Disney and ABC, and our animals appeared in it. Our trainers were essential to it. And of course, when we had the producers and the director and all coming in and they're looking at this, oh, this is perfect. Perfect. This is so organic. And so they shot it as it is. They did change our name, but they've given us a retainer because if this gets picked up, they like to use it permanently. The show as being the main character's home base out of our Marine Mammal Center. Along those lines, been spending time chatting with the Pacific Wildlife Foundation, with Rob and Rod about expanding our mandate and doing things also as a marine science center. So not just for marine mammals, but for those who haven't been there, this is an amazing piece of wildlife that we have here right next to Vancouver. It's hard to believe we are literally in Vancouver, but we see the ability to do a lot more and to have a truly floating research center that not only does science and does environmental monitoring, but also does education and can have kids come down. As an example, I know one of our summer students this year was quite surprised that she worked for us and she's on the track team, and then she wrote a piece that some of you may have read about sort of coming to UBC and one of the motivations, and it was because when she was a kid, elementary school, she went down and saw our sea lions. He never told me that. I had no idea. So I think we can affect kids, adults, people. We've got an amazing place to do it. So hopefully we've got 6 months of life still, reaching out still to give us an extra 6 onto that, and hopefully 2 years onto that, and get us through. But we are at a major transition phase, so stay tuned. Thank you very much. I'd be happy to take any questions if anybody wants to while Brittany comes down and gets set up. So, Brittany.
Yes. All right, good afternoon. My name is Brittany Vasona. I just finished my master's degree at the University of Aberdeen, Scotland. I'm going to present some of my master's research that I was working on. So I was looking at the effects of schooling characteristics of pelagic prey on marine mammal distribution and abundance. So when we look at studies on marine mammal distribution, they basically kind of focus on where predators are in relation to prey. This is mainly done by looking at the presence, absence, or the density of marine mammals, and then comparing that to prey biomass. And if that is not available, they'll use a proxy, such as an oceanographic variable, to indicate the level of biological productivity in the area. But there's not always a strong relationship between the spatial overlap of predators with prey biomass. There's other factors, things about the prey patches that are shown to really influence when and where you're going to get this overlap of these predators with their prey. So things like the patchiness, as well as the location in the water column. So these are things that we should also think about when looking at marine mammal distribution. So when you think of marine mammals and prey patches, it's usually in the form of fish schools. So fish exhibit a wide variety of schooling characteristics, everything that will affect their morphology to their orientation in the water column, thus affecting their availability to many predators. So this kind of got me thinking of how these schooling characteristics are going to affect the distribution and abundance of marine mammals. So in a completely different location, my study was in the North Sea off of the east coast of Scotland, a region called the Firth of Forth. So this region has a lot of foraging marine mammals. It's home to gray seals, harbor seals, as well as harbor porpoise, and also to sand eels, which are also known as sand lances over here, but they are a pelagic schooling fish, and they are the main prey to these marine mammals. So these were my main species that I was looking at. So my data that I used that came from the marine mammal was density distributions and sea usage maps. So basically the brighter squares is a higher density of animals in those locations, and these are kilometer grids, so you would get higher individuals in the brighter colors. And I pulled this information from the Marine Scotland Interactive database for grey seals, and they also had one for harbour seals, so different distributions. And then for harbour porpoise, but this came from a different survey. This was the SCAN survey, so it's conducted in 1994 and in 2005, and this is a year survey that you get the density of harbour porpoises in that year. For the information about the sand eel schools, they conduct annual acoustic surveys in the Firth of Forth. So I used 2008 to 2015 data, and basically you have the boat will go over, collect acoustic information, you can pull out all this information about the schooling characteristics. So I took that out by year and averaged it so that you get the average schooling characteristics in those same grid cells in relation to the marine mammal densities. So I set out to kind of answer this question, if the density distributions of gray seals, harbor seals, and harbor porpoise could be explained by these average sand eel school characteristics. I did so using statistical models, so I used generalized least squares models where I had the response variables being my densities of the marine mammals. So I ran 4 models with the explanatory variables being those schooling characteristics. So I looked at school density, school length, as well as school thickness, compactness, school depth, and the number of schools, which was an indication of patchiness. So from these models, the main kind of findings that I found were that for each species, the best models to account for the variations in densities, the best schooling characteristics to account for the variation in marine mammal densities differed between species. So they had different combinations of schooling characteristics in the final models, but all models only contained school density, thickness, compactness, and school depth. Of those, school depth was not significant by itself. Only in combination with the other schooling characteristic was it significant in accounting for the variation in these marine mammal densities. And for each different species, they had different characteristics that were in each of the models, so this is likely related to their foraging strategy. For example, in the final harbor porpoise model, they had the thickness of schools, so their vertical distribution through the water column, as well as when they were most dense. Those were the main 2 properties when you would get the highest number of harbor porpoises in that particular area. But in general, the predators favored these schooling characteristics with the greatest spatial-temporal predictability, so that were the most common characteristics over space and time with the least variation. So kind of the take-home message that I got from this was that the schooling characteristic or variations in marine mammal densities can be explained by these schooling characteristics. So it's something else to kind of think about when you're looking at when, where, and why we get marine mammal sightings. But these schooling characteristics are driven by biophysical variables. So we can determine what those biophysical variables are that are creating these favorable schooling characteristics, and when and where we're getting these marine mammal sightings, we can start to figure out in what areas these prime conditions are to better place our marine protected areas. And that's pretty much it. So thank you.
Any questions? So did I pick up right that predictability is key or not?
Yeah, so the predictability of where these patches were going to be, kind of for the variables that were in the final models were the ones that showed the less variation in them and that were kind of the most predictable in that particular area, and that's where we were seeing the most sand eels in over all the survey areas, as well as where most of the marine mammal sightings were too.
Any other questions? I lost track of the time. This is why you should never chair something when you actually give a talk. I realize we went past our 2:30. I'm going to suggest we're going to take our break now. If that's okay with Dom Tollett. Dom says yes. Okay, so we're going to make it a 30-minute break, which will take us to 10 minutes after the hour. So 30 minutes from now, and we'll be right on track. Thanks very much. Oh, and could the speakers in that last session come down in case people want to chat with them? Okay, welcome back to the BC Marine Mammal Symposium. We're now down to the last 45 minutes. And then you can talk as much as you want. A couple people asked me just about Stellar IPA. Where can I get it? Well, I know that I had to special order it from the UBC store over here. So I know that some of the stores are carrying it, but it's not widely available yet. It's available in the United States and in Canada. Although I also learned that we've got viewers right now from all over the world, and we just surpassed 1,800 people watching. So it's numbers have gone up all day long. Our next speaker is Dom Tollett. And talking about noise.
Thank you, Andrew. Thank you, everyone, for sticking around. I am a marine mammal consultant. I do risk assessments. I actually also teach elementary school for a great program called Scientist in Residence. The company I work for, Smooth Consulting Canada, is owned by the University of St Andrews. And I'm going to start with my MRU graphic, just highlighting all the sort of uncertainties involving risk assessments in terms of sort of likelihood and magnitude. You know, fishing should not be ever off the dialogue table when you're talking impact. And risk assessment is all about looking at the vulnerability of a species to start with. Fishing industry, clearly, number one, obviously, culls, whaling, hunting, high magnitude, strike, a really important thing for large whales. We haven't talked today about the Navy and their war games and use of sonar, but clearly mortalities due to those actions. Same with the seismic noise industry. Oil spill is a problem that doesn't go away because of human error and malfunctions. Pollutants, one of those chronic, often chronic problems ranging from plastics to PCBs to fire retardants, thousands of different chemicals coming to the market. We've obviously got infrastructure and vessel noise, pile driving, different types of vessels, operational noise from wind farms, wave and tidal structures. And then obviously overlaid is, I'm not quite sure where to put pathogens, but obviously potentially high magnitude and you, whatever Trump might think, environmental change is happening. There's going to be winners and losers with marine mammals, but the sort of final sort of take-home message, this is risk assessments are complicated, and we've got a major management responsibility for understanding and taking action, especially on some of those high magnitude effects, as well as the high likelihood stresses. So I'm going to go through a few slides talking about risk assessment, and risk assessment doesn't just stop with a risk assessment, but often moves to a monitoring phase as well as a mitigation phase. So I'm going to be talking about some of the projects we've been involved in this year. The first one is trying to improve risk assessments. The bottom line is people are trying to get better at risk assessments. They are not easy with marine mammals, far from it. I think number one, they need to be transparent. At the moment, they're sadly not. And what we're trying to do at the moment is include some of this biological and environmental context, as well as species vulnerability, and as well as uncertainty within those risk assessments. And the next stage that we're working on is actually trying to incorporate the hardest bit, that sort of long-scale chronic effect of human activity. I've been on the same topic working with collaborators, mainly at DFO Science, and this paper's just come out. And it looked at all the behavioral response studies around the world. We looked at 370 different studies. In the end, because of inconsistent reporting, we were down to just 80 studies that we could look at how marine mammals respond in a behavioral way to different levels of sound and their severity of response. And I think the take-home message at the moment is we should not be relying on the current generic receive level thresholds, such as the 160 dB that is used for multiple species. The bottom line is the severity of the response is driven by the sound source and the hearing sensitivity. And there's obviously a need to be consistent in the reporting of these behavioral response studies, those people that do this work. Next up, again on noise effect assessments, we ran an expert elicitation for a population consequence of disturbance model for NOAA on Cook Inlet, Beluga, and noise effects. And the reason you need to undertake expert elicitation is this massive knowledge gap between behavioral disturbance effects and effects on key vital rates. And until the work of Lance and John gets into the mainstream, there is still this big gap, and this leads us to the development of these interim PCOD models. The interim because until we've got real data, we're asking a group of experts what they think about disturbance and the effects on vital rates, such as lactating females, the production of calves. Those sort of questions are what we're asking. And then we try and fit that into a model that we can model long-term population viability. And there's a report on our website if you're interested in PCOD. In terms of noise effect monitoring, we've got a new calibrated hydrophone at Limekiln Point State Park on San Juan Island. It's live streaming. There's the link there. It's deeper than it's been before, which was considered important by the acousticians. And the aim here is largely to collect long-term ambient noise data. This is a collaboration with the Whale Museum and Port of Vancouver Echoes program, which is aiming to better understand and manage impact of shipping noise on at-risk whales. So this is a key site for southern residents, and we're interested in understanding how management actions like slowdowns or changing propellers or defouling might reduce noise levels for killer whales. Also on noise effect monitoring, we've been working at various tidal turbine demonstration sites, including the Force site in the Bay of Fundy. And here we've been using C-pods, these click detectors on the mid-scale range, but everyone's interested in avoidance and movement around these tidal turbines. And the strategy we've come up with is using active high-frequency sonar. We're using the Gemini. It's 720 kilohertz frequency, so that's 500 kilohertz higher than the echolocation of porpoises, which are the high-frequency species. So we did various tank tests and field tests showing that marine mammals don't respond at this level of frequency. We've been using sonar both outwardly facing as well as looking at the turbine, coupled with an array of hydrophones from 4 to 12 hydrophones placed around the turbine. And you can see here, here's the turbine top left. It's called the open hydro turbine, 16 meters. Ruth Joy will be talking a little bit more about the work that we're doing to look at how data from these studies can feed into avoidance by marine mammals and improve strike risk models, with the overall aim, I guess, is to de-risk an industry that can provide renewable energy into the future. This turbine here is now grid connected, and 500 homes are now being powered from renewable energy. In terms of mitigation, one of the studies I worked on was industry-funded collaborative review of mitigation monitoring technologies during low visibility. So during the day, on a nice day, observers can detect marine mammals, but during the night, the sea state increases. If it's foggy, things get harder. And so this was a study that aimed to look at the actual performance of different technology, passive acoustics, sonar, radar, and infrared thermal. And I guess for me, and this was focused on the seismic survey industry. One of the take-home messages was how poorly passive acoustics performed for detection of whales during surveys, and how this new infrared thermal method might be the perfect concurrent method to use, as well as PAM and observers. And I point you towards the Zitabart publication on thermographic videoing. It seems very effective, mainly in temperate and Arctic waters. It may not work in tropical waters because it uses temperature difference, but some interesting results there, and that study's complete. And lastly, this is a new product we've been working on. I presented in the past talking about large ocean-going passive acoustic monitoring buoys. This is what we call a coastal acoustic buoy. It's just 45 kilos, and it's aimed at mitigation monitoring, for example, pile driving. It's built by the Applied Physics Lab in University of Washington. So it's small, 2 weeks battery power at the moment, and the aim is to have automated real-time cetacean detection. We've got a range of about 2 kilometers that we're hoping to improve. And this is already being used in Hong Kong. And this is something that if you're interested in, I can give you the contact of the lead scientist there. And I'm going to just end it there. Thanks for listening. Check out the website. My email's down there. Thank you very much.
Do we have any questions for Dom?
Thanks, Andrew. No, I'm sorry, I probably missed this, but that mitigation monitoring buoy, is that does that accomplish mitigation by making sure that there aren't marine mammals around when noise is generating, or does it measure noise?
It does both. So you've got PAM guard running internally that is a whistle and moan detector for dolphins and whales, and then a click detector for echolocating cetaceans. But then it spits out various noise metrics.
I guess the reason I was a bit confused was because you said that passive acoustic monitoring isn't a great tool for use, or you've been finding that it hasn't been for monitoring marine mammal or detecting marine mammals during surveys. I've always worried, as I think you know, I've always worried about passive acoustic monitoring, but anyway.
That was, we did an analysis of all the seismic surveys, sort of PAM versus MMO reports. And so that was specifically having a PAM line being towed behind a seismic boat. So your seismic boat is booming every 12 seconds. You know, you're perhaps the most hated person on the boat. So they don't think about where the system might be trying to what it's trying to do. So you're the least priority of the whole project. So there's different ways of improving it, but basically at the moment, yeah, towed PAM behind a seismic vessel is not doing a good job at picking up whales, and that may be partly, and there's evidence to show this, that the whales shut up because of the seismic survey. So it may not be just because PAM performs badly, it's because the whales are affected by the seismic.
You were saying infrared thermal is something that looks promising for detection. And I'm just wondering at what sort of distance?
About a kilometer it depends on the size of it uses the blow mainly rather than the heat of the animal. But the reliability to a kilometer was actually surprisingly good. Yes, the one of the recommendations was you need to see if this actually works in tropical environments. I suspect it's going to work.
Can you briefly comment on the importance of acoustic monitoring for the effect of, for example, traffic, ocean traffic, and exploration noise on ecosystems as a whole? So impacting prey species for cetaceans that depend on other animals, not marine mammals?
Good question. I guess the best answer is there's an acoustic conference every year. This year it was in Dublin, and the number of presentations that we saw this year that were on fish and cephalopods and invertebrates was 10 times higher than 5 years ago. So there is interest there. Obviously, as you're monitoring ambient noise, you can make that crossover in terms of effect once you understand what levels might be causing effect. So there's definitely science going on. It's fairly nascent, I would say, at the moment, but it's definitely growing as well. I think there's a real understanding that that's a data gap that needs filling.
I'd like to echo suggestions that I've read and heard. Stealth planes and submarines run very quietly. Is any of that technology, such as noise canceling, being applied in these high noise production traffic areas?
There are vessels now that have that sort of capability to run less noisy. The ECHO program and Port of Metro Vancouver have actually given incentives to boats that use those technologies. So it's sort of starting, but no, there's still plenty of room for improvement on using those technologies. Some adaptions added to the propeller, reduced cavitation. There's not a lot of science there at the moment. There's a lot of proponents suggesting that they might work, but no data yet. So that's one of the good reasons for sort of setting up these different nodes for monitoring. In fact, the port has got a underwater listening station in the Strait of Georgia that is monitoring ship noise and can look at how much of a difference did it make if I added something to the propeller to reduce cavitation. So the science is starting to go there. In terms of how much it's being used, not very much, but that is the future, clearly, designing vessels that are quieter.
Stop there. Thanks so much, Dom.
No worries.
Now on to, not just with joy, but the topic, tidal turbines.
Hello, everyone. Thank you for sticking around. Dom went into sort of superficially talked about a whole bunch of projects, and I'm going to dig a little bit deeper into a project in Northern Ireland that we worked on together, and it was the Strangford Lough tidal turbine. And this is a picture of the turbine. It has 2 rotors on it. Each are 16-meter diameter, and this is with picture of the rotors out of the water. Of course, they go in the water to actually generate power. And that's when they become dangerous to, or possibly dangerous, pose a risk of being a danger to marine mammals. And so what this project was to try to quantify that risk by tagging some harbor seals. And this turbine stands at the entrance to an inland sea, and this inland sea is the biggest inland sea in the British Isles. It's 150 kilometers squared. It has some massive tidal exchange of water, so I'm not sure how fast it goes, but at least over 4 meters per second. And the turbine spins both ebb and floods. And so just to give you some context, because this has been in the news lately, this is a picture of the Bay of Fundy force. Dom had actually a better picture than that one. But it's the same diameter, so that's 16-meter diameter. And so the first one went in 2 weeks ago, one of the 5 test sites in the Bay of Fundy. So we don't have any data yet back from that. So this talk is about the Strangford turbine that was deployed or constructed in 2007 and 2008. And they had the wisdom to put the tags on the seals both before the turbine went in and after the turbine started operating. And so this is a slide which I'm sure you're all reading closely. And it's to emphasize the point that we all love math, and particularly math that's related to marine mammals. And of course, risk changes over time, and it also changes depending on the structure that you have underwater. So how fast those tidal blades are spinning, and where in the water column the turbine is located, as well as a bunch of biology related to the seals. So how fast those seals are swimming, and that changes too over the tidal cycle, what their behavior is at various different points in the ebbs and flows, and how that changes over time. So that's what we were mining this tag data for. And so the Sea Mammal Research Unit instrumentation group at St. Andrews put these phone tags on, GPS phone tags, and they also recorded the depths of the dives, but because they were communicating with cell phone towers, they had a thin amount of information that they could transmit. And so each dive was represented by 9 points equally spaced. We don't have the kinds of data that Hassan was presenting with, I don't know how many points per second, but it still gives us a good dive profile for harbor seals around a turbine. And so what we were interested in is looking at where the turbine is relative to where the seals are fishing. And if you think of some of the presentations we saw today, I think Hassan showed them harbor seals diving down to 300 meters. And I'm not sure how deep the tanks are at the aquarium, but I think this is sort of like blasting a high jet of water at seals in an aquarium, because it's only 24 meters deep where this turbine is. So they're constricted in depth, but they have to swim, catch food, and navigate a system that's extremely active. And so obviously getting to the bottom of 24-meter depth is of no problem at all. And for these seals, these were extracted dives from 200 meters with distance of the turbine. So this is in the middle of the channel. So this is at water speeds that are maximum for that body of that inland tide. So these seals are not overlapping where the turbine is located in the column. And you can see it doesn't change pre versus post turbine. And because we have GPS tags, we can build movement models and spatial maps of how they're using the channel. So that's the 200-meter radius circle there where all the dive data was extracted from. So that's in 2006, before the turbine was built. And this is the data from a set of seals. So there's 12 seals tagged in each of the before and after. And so that's the surface that's being built post-turbine. And this the tag data stretched from April through to the end of July. So we don't know much about what they do in the time. And that's all I have to present. But one thing I meant to say that's interesting about this from a historical perspective is that the very first tidal mill was built here in the 700s by the Irish monks, and they used the power of the tides to grind their corn. So anyway, thank you for listening.
Question up the top.
Has there been any acoustic repulsion device devised to any animals or creatures away from these
Well, the turbine itself makes quite a bit of noise. So it's at 1 meter, it's estimated to be 175 decibels for the Strangford Lock turbine. So Gordon Hasty, who used to be at the Marine Mammal Research Unit, estimated that it should be audible at 500 meters. So in terms of an acoustic deterrent, it sort of is its own acoustic deterrent. Oh, he's asking, is there some kind of seal pinger or an acoustic deterrent that could be used in addition? And I'm not qualified to answer that, but I presume you could put something on if you needed it. But as we've shown in that spatial map there, it's doing a pretty good job of keeping seals away from the turbine.
So Ruth, in that last figure you showed, is it possible to turn the turbines off? See if you get a response where the seals come back? Can you be sure that that's the effect of the turbines?
Of the actual rotor spinning as opposed to the presence of it? Yeah. No, and we can't be sure. There's other reasons why they may have not been there, and that is the redistribution of prey as a result of the removing that power from the stream, and also the sedimentation that happens as a consequence of putting a turbine down. So it's at this point, I don't think the technology or the knowledge about underwater turbines is at a state where we can speculate. But yeah, good question.
Nothing simple.
Yep.
Not even the math. Oh, Dave's got a question.
Okay, so just to give us some sort of, for me at least, some concept of how fast these turbines, either the one in the Bay of Fundy, which we might be more concerned with, although that's a solid turbine, isn't it? The Bay of Fundy?
Yes, with an annulus, so there's a...
Okay. So that's not as much of an issue as these giant propeller turbines. Like, how fast are these actually turning? Like, you know,
so that one turns 14.3 revolutions per minute. And not having actually seen it rotate myself, I imagine it's something like the Grouse Mountain turbine. They don't rotate that quickly. In fact, people claim that because they rotate so slowly, they bear no risk whatsoever to marine life. Whereas
I mean,
others,
it's not. I think a lot of people, when they think of turbines, the issues turbines, are thinking of, you know, scene in Indiana Jones where the guy gets stuck in the airplane propeller, but it's not moving that quick.
No. In fact, the Strangford Lock was engineered to feather its blades so that it at a certain speed, it was at maximum rotational speed, even though the water was going faster. So it beveled the blade angle so that it sloughed potential power, and that was partially because of the strain on the system. If the whatever stuff that works behind that generates the power couldn't operate at full blade capacity.
So I want to thank Dave Rosen for sharing his graphic visual with us, and Ruth Joy for a nice talk. Thanks, Ruth. Now, Michael Harris. More presentations, and then a couple of announcements to wrap up.
Work? Good. Hi, everybody. So my name is Michael Harris. I am the outgoing executive director and spokesperson for the Pacific Whale Watch Association. And this is Tasley Shaw. Tasley is with Steveston, did I say that right? Steveston. Yeah. Seabreeze Adventures Whale Watching. And this is our panel. We're a two-person panel. We're supposed to be a three-person. We lost Casey Brandt, Dr. Casey Brandt. We do have a telemetry tag on her. I think she's in Victoria somewhere. Somewhere off, though. Yeah, anyway. So this is the fourth time I've been here, and I love it. And I really thank Andrew for inviting me and your whole team, Pamela. It's been a lot of fun. Like I said, I am an outgoing executive director, not unlike our outgoing administration. I've heard a lot of great wonderful projects. We have a lot of great projects, conservation and recovery projects on the US side. With this new incoming administration, he'll probably throw everything out and replace it with something really terrific. So anyway, how's that for depressing? No, I'm kidding. So I'm going to kind of go through this really quickly. I know we don't have a rubber chicken this year, so I was really fearing the rubber chicken. But I'm going to kind of scoot through this, and I'm going to kick it to Tasley. Operate this way? Down. All right. Well, first of all, what is Pacific Whale Watch Association? We are the commercial whale watching industry in British Columbia and Washington, at least on the inland waters here. We are now, the association is now up to 37 companies operating out of 21 ports. The association is now conducting over 14,000 whale watch trips each year, with a fleet of roughly about 85 vessels within the 37 companies that operate out of these 21 ports. Growing at about an 8.3% or higher clip a year. And I'll explain a little bit about that growth here in a little bit, but we did an economic survey a couple years ago where we actually opened our books and did a blind study with a CPA in Victoria and with the University of Washington, a couple economists at the University of Washington, because there's never been a transboundary economic study done of whale watching. It's always been parsed out to Washington or BC. And what the economists came back to show us that in 2014 that we actually contributed $144 million US in economic impact just in 2014. And again, showing a growth of about 8.3%. We are growing, the association is growing, whale watching is growing. And now we can quantify exactly what we are out there and what we're contributing. So I'm going to go through briefly. I've got a little PowerPoint here. Again, it's a lot of slides, so I'm going to scoot through some of this. I want to leave time for question and answer because I think the last 3 years I've been here, I kind of ran way over time. We didn't have any Q&A. So I'm going to kind of scoot through this. I can just do sort of this overview of some of the encounters that we've had in the last year, which have been really exciting, really interesting, really alarming at the same time. And I'll explain that in a little bit. Some of the media related to this. And then kind of where we might go, where the association might go from here. So scooping through here. I'm going to play a quick video here. This is what we started calling the Saratoga Grays. And this is, we've got a group of gray whales that in the last 25 years, John Calmakitis and Cascadia Research Collective have been documenting these whales that have been coming off the Great Migration, coming into the mid-Puget Sound area and some of the very urban areas, feeding on ghost shrimp each spring. And they are here like clockwork. In fact, we have some that have showed up every year for 25 years. So it's a really interesting group. I think it's up to 11 animals that regularly visit Puget Sound in the springtime, eating ghost shrimp. And but I'm not sure how many total individuals they've counted over the years, but I'm going to show you a little clip here.
Researchers attach a camera to a gray whale in Puget Sound. Nice job. Providing a rare point of view look at one of the most epic migrations in nature.
This is really the greatest migration on the planet Earth.
Every spring, 22,000 gray whales begin their migration from Alaska's Bering Sea to the lagoons of Mexico. But starting some 25 years ago, a very small group started making a 200-kilometer detour into Puget Sound.
Because they learned that there is just a bounty of ghost shrimp in this one area.
The area in question is called Saratoga Passage off the waters of Whidbey Island, where the whales take a big risk to fill their bellies.
Well, when you think about a gray whale having to come into water that's only 6, 7, 8 feet deep, that's barely deep enough to allow its body to be in there. And if it mistimes that, tide goes out too soon, they could get stranded.
But with only a few whales out of thousands making the trip, it begs the question, why don't more whales make the journey? Well, it would seem that, like all good fishermen, these whales, usually the same ones every year, are not about to share their secret fishing spot.
And obviously it's not being widely shared. It's not a very big area. This is really a pretty small area we're talking about, so it couldn't support very many gray whales.
Here we go, 1 o'clock. Here we go.
But the ones who do come put on quite the show.
Well, right there, right there, right there, right there.
delighting whale watchers who are willing to brave the choppy waters and cool winter air.
Oh my God!
It's a sight to see, but it's also a very promising sign. 20 years ago, gray whales were endangered, but they have rebounded, and officials think it's reason for optimism for the local population of resident orcas.
We can get these gray whales to recover from from where they were. We feel pretty good about the southern resident orcas.
The gray whales are expected to be in Puget Sound for about a month until the shrimp runs out before rejoining their friends for the long journey to their southern spawning grounds.
And yes, people do get excited about gray whales. I'm saying. Anyway, which is funny because it is now. It's such a unique and an easy, frankly, very easy commercial whale watching operation. They're right off there. It's 100% guaranteed that we're going to see them. And people just love to see them. Don't you?
Oh, yeah.
Oh, yeah. All right, skipping through here. Interesting, because they're starting to come down here, and because we've had such, and I know that Joan Lopez mentioned about the transient frequency we've had, the big whales that we've had in here. Well, when we're getting gray whales in here, we're getting bigs in here, we get a clash of the titans every once in a while, and that is something that the media loves. And part of what I do is I'm a producer for ABC News, and I know that we like anything conflict, even if it's in the wildlife, and even in the wild kingdom, we like conflict. So it gives us an opportunity, again, just as I'll talk about later, about spreading the fleet, making us more than just orca watching. Here, we're watching all whales. So skipping through here. All right, and we're being picked up globally. I mean, just little things like that. Just a brief encounter with transient killer whales and gray whales, which didn't really amount to much of anything, frankly, just got picked up globally. So media is definitely into it. Okay, so we had a little bit of an abbreviation in what we were doing because of L95. And this, I'm going to scoot through here a little bit, but as many of you know, we had L95, a southern resident whale that washed up with, I believe they were titanium barbs still in the site where it was dart tagged. And so the Center for Whale Research, along with the Pacific Whale Watch Association, decided to make a very strong statement about this. Actually, Ken Balcom a little bit stronger than I did. But making the point basically that, we need information, we need to track these whales, we need to fill in these gaps in research, but let's look at the methodologies we use and let's not be invasive. So anyway, scooting forward here, I'm gonna go back to this, but we called for NOAA Fisheries to just take a break, figure this out, study the methodology, figure out how we're doing this, find a less invasive way to do this, and thankfully, they did take a break. They took a break and went back and have been analyzing the efficacy, but also just the invasiveness of the methodology on the tracking. So I'm going to scoot forward, though. One of the goals that I had in my 5 years, I actually started on this path about 5 years ago when I was asked to start consulting the association. I wanted to spread the fleet. I didn't want the association to be thought of as, the entire fleet from 21 different ports all converging in one area and on southern resident orcas. So we started looking at kind of what was going on out there and some of the other whale watching opportunities beyond the gray whale, watch off, off of Puget Sound, but looking all around. We knew that there's a lot going on out there. And I will get back to this picture, this amazing picture, but that happened right off Victoria last August, I believe. But we also have had, as Joan Lopez mentioned earlier, and I'm sure others have addressed here today, we've had this year after year of a record number of sightings of Biggs killer whales. Very interesting whale watching, obviously far less sensitive than southern resident orcas. But we had this opportunity to start exploring other ecotypes, I guess this other ecotype of killer whale. And there's one way to spread the fleet. And then we've had in the last few years, as you know, and you've probably addressed here, the humpback comeback. So I'm going to play a quick video here.
Seeing whales in their natural habitat is something West Coasters sometimes take for granted. But there is something special happening in the Strait of Georgia right now. that is thrilling even the most jaded whale watchers. Kylie Stanton has more on the spectacular humpback comeback.
Just on the Canadian side of the border, I heard on the radio that there were upwards of 10 to 15 more spread out along this line.
It's not just a matter of luck anymore to catch a breach or count the blows. On almost every trip, they're putting on a show.
Oh my goodness me.
Call it a humpback comeback.
Yeah, it was really ginormous.
It's humbling for most people. And I'm told just from a boat that just got back that there's probably 20 to 30 out there right now. So,
In recent weeks, whale watching crews have reported unprecedented numbers of the massive acrobatic whales. Look at that blowhole. Not just 2 or 3 at a time, but instead large groups traveling together.
It's basically the whole seascape filled with breaching, foraging, peck slapping, tail lobbing. It's humpback heaven.
They're congregating up and down the Strait of Juan de Fuca, off the coast of Victoria, all the way to the Olympic Peninsula. It's painting a very different picture from just 50 years ago. When whaling was banned in 1966, there were just an estimated 1,600 humpbacks around the North Pacific coast. That population has now exploded to about 21,000. But what's behind the recovery is a little more complicated.
So it's an overlapping series of events, I think, that are bringing these North Pacific humpbacks into the Sailor Sea again, not just their increased numbers, but because there's possibly some issues out there on the coast and the rest of their habitat, and they're finding fish in here.
Unlike orcas, whose main food source is salmon, humpbacks are opportunistic foragers, feeding on krill, sardines, anchovies, and herring. Their flexible diets may be playing a big role in why they're doing so well.
Bring the jackets.
Regardless, it's providing once-in-a-lifetime opportunities for researchers and whale watchers alike.
I'm like a kid in a candy store. It's fantastic.
Kylie Stanton, Global News.
Hope you had a good time.
Victoria.
Thank you for that.
We got this humpback comeback. As you all know, 10 years ago, we started seeing them coming in one by one or so, and this year has just been crazy. We've seen groups of 50, 60 off of the south end of Vancouver Island. We originally coined the term humpback comeback, and this year we started calling it humpback heaven. But the one thing that we didn't want to do, well, I should go back to this. First of all, what we realized was that there is just a huge amount of people around the planet who want to come see humpbacks. And so after we started advancing some of this humpback media, and again, spreading the fleet and talking about other opportunities to watch whales and other species of whales, we started seeing it getting picked up globally. We started seeing literally hundreds of millions of people around the planet paying attention to what's going on with the humpback comeback here in this part of the world. And now we have, it makes up a vast majority, I won't say vast, but definitely a majority of our whale watching trips are humpback whale trips. And nowadays, people come to the docks and say, we want to see humpbacks. Whereas a few years ago, they just wanted to see orcas, or they wanted to see southern resident orcas. And there's this kind of this pressure on the boats and the fleet to kind of go see southern resident killer whales. And so this humpback comeback, if you will, is really giving us a chance to take some of that pressure off the southern residents and really spread the fleet. And honestly, it's been a boom. It's been a boom for Pacific Whale Watch Association, and we're very pleased. We've even learned to identify some of these animals and start talking in the media about them in a very familiar way. And again, here it is being picked up just globally. People are really into the humpback comeback here. But the one thing that we didn't want to do is just say, as you know, every time we did a story on humpbacks, people would always ask me, and I'm the only, probably the least qualified out of everybody in the association to actually explain why they're here. But I didn't want to just gloss it over. I didn't want to just say this is a great, wonderful thing that's happening here. I wanted to have the media encourage people to ask questions. Why are they here? And advance studies to see, why they're here and are they impacting, how are they impacting the ecosystem? How are they impacting prey resources for killer whales, for example? And so one of the things we did is one of our captains, a 20-year captain, Rhonda Reedy, is proposing to do a PhD study on what are they eating and how are they impacting the ecosystem here. And so we've been supporting her, and she's been getting support elsewhere. And so we're real excited about trying to answer some of the questions. Why are they here? What are the impacts really? for this humpback comeback. Pushing through, we had a very unusual event in July here, common dolphins. Now, we're not real clear, and I don't think even Cascadia is at this point, whether they're long-beaked or short-beaked. We just knew that these common dolphins are not common here. And so we punched this story out and started sending photos out and started working with Cascadia to try to identify them. And whilst we were doing all that, they merged the two species, didn't they? They long-beaked and short-beaked, and now they're just one species. So it kind of rendered that moot. But we had a very unusual sighting, and they actually hung around. The school kind of hung around and made their way into the South Puget Sound sometimes. I saw them off Seattle waterfront. Very unusual, very interesting. But again, why? You know, we didn't want to say just this is a cool thing, isn't it? We wanted to say, well, there's got to be something behind this, and it causes some concern. As I said in here, I said it was exciting and alarming. This is highly unusual, and I guess in the best of both worlds, they really shouldn't be here. They should be down south, but we had them. More pictures. These were taken by Jim Maya off of Victoria. Again, more coverage of that. And now, we had what I was put a question mark on, because I don't know what that means, but it's the second fin in. Last year, not this year, but last year, at least in the Puget Sound area, and we have sort of our boundary for Puget Sound is kind of Smith Island south, but we were getting a fin whale last year, a juvenile fin whale, not this guy, but a juvenile fin whale, which was the first confirmed sighting of a fin whale in the Puget Sound region since 1930. Very, very unusual. I'd like to put an optimistic spin on it. I'd like to think that fin whales are coming back. As a matter of fact, what you're seeing here is the adult that we had this year coming in, roughly the same area, more toward Port Angeles, Port Townsend area. But again, is this, you know, as I said in the media, it's like finding a brontosaurus in your backyard. This is really cool, second largest animal to ever live on the planet. And so we had some opportunities to talk about, well, you know, two of the whales that got really targeted by commercial whaling here off Vancouver Island were humpbacks and fin whales. You know, humpbacks were extirpated generally by commercial whaling, and they have come back. So maybe this is the fin whales are recolonizing. So that's my hope. So we moving along here. Very interesting. Here's another one of those, you know, wild kingdom conflicts that the media loves. But had a little bit of a very interesting event happen off of on a rainy day off of Victoria. Back in August, I think it was. Was it July or August? It was August, where we had humpback whales seemingly intervening in a stellar sea lion hunt by big killer whales. So we had, you know, that was a fun story to get out. It was very interesting. A lot of speculation. We don't know exactly what happened. But a very interesting story that got picked up very, very widely around the planet. We got great photos. These photos were largely taken by Russ Nick's BC Whale Tours, but we had great assets, great photos. And that's the thing, when we're out there, we have really good photographers like Tasley and like Casey and like, you know, there's a lot of good photographers. So we're getting a lot of images that we're sharing with researchers, and we're just contributing to conservation science whenever we can, trying to help scientists like you all understand what's going on out there. Editorial from Times Colonist. I really like this because I like anytime we get the word hope or hopeful into any discussion about these whales. There's a lot of gloom and doom, and we're going to talk about that a little bit, but so I like that. I like the fact that we, you know, we're able to talk about these poster children of species recovery with the humpbacks, North Pacific humpbacks, with gray whales, of course, North Pacific gray whales. It's very cool to be able to talk about that and to witness and see these animals in the wild. And we are contributing to a bit of a boom here going on in Vancouver Island particularly. And of course, that's contributed by contributing factors, of course, is your Canadian dollar, of course. But whale watching is really driving the economy in more ways than I ever imagined when I got started 5 years ago as executive director with these guys. Very excited. And as I said, it's been the year of the humpback comeback, humpback heaven. Footprint. One thing I've really tried to urge the whale watchers over the years is that we're in a unique position as stakeholders and with the expanse that we have and the resources we have to really reduce footprint on the fleet out there and to try to advance some good whenever we can. So I'm going to scoot through this a little bit as we can, but talked about a tidal energy turbine project, Admiralty Inlet, a couple years ago that we had some concerns about. Again, as our headline says, the right project in the wrong place. But we said, you know, I think these guys need to go back to the drawing board because what they've got now is just it's an issue. And so we stood with the treaty tribes of Washington State, who were very concerned about the impacts of the pilot project as it was going to be installed. We contributed to helping them rethink that project. You know, and I'm just going to scoot through some of this because this isn't a demonstration of our great PR. But one of the things we did too is that, you know, we're looking at we teamed up with Ace Hardware stores, 4,700 stores, started small, trying to do orca-friendly products, not unlike Dolphin Safe Tuna. I have some friends who were back in the early days of Dolphin Safe Tuna. It started as a good idea and started in a few places and then grew big, and we hope this will grow big someday soon. We're helping to work on projects to get fish in the water, real hands-on projects. This is the Sook Chinook project. I call it project. But that's one of the things that we're doing. We're involved with a lot of salmon restoration projects and supporting them and getting our hands dirty on some of them too. And then what I call a damn good idea. I love dam removal, antiquated dams. And this is a Klamath, which historically was the second largest salmon-bearing system for southern resident orcas. And long overdue to get those dams out and get that thing going by 2020. We hope that we're going to start seeing some real fish. We have some great returns already on Elwha and some other places. Why not here? So we're excited about that. We've helped a little bit on drones. Number one, having a policy among us that we would really not have it as part of the whale watch experience like they do in California. A lot of California whale watch boats really will fly drones, sell videos to passengers, make it part of the whale watching experience, and we said, no, we're not going to do that. And we've just sort of helped enforcement put a clamp on that. I think last year for Christmas, there were 1 million drones given away in the United States as Christmas presents. So there are a lot of hobby drone flyers out there, and it's an issue. Also, you know, I said it's a good thing we're out there. We're helping because we have a fleet out there, and we've got so many boats out there, and we have some good expert eyes. We can do things. This is an entanglement of T77A. Had some fishing entanglement. We've had other cases where we have seen, or Paul Cottrell from DFO has seen entanglements, and he's asked us to kind of follow up and just see if we can report and see. In this case, we're able to confirm that the entanglement on T77A was shucked somehow and that the whale was fine. We've been involved in a number of rescues on the water. You know, I mean, that's kind of the stuff that we should be doing out there. We should be helping whenever we can. And this just came out, I think, yesterday, and this is from Eagle Wing, Casey Brandt's company, you know, speaking out on the Kinder Morgan project. So we're trying to make our voices heard whatever we can, and I think our voices are getting picked up. Now, Meltwater Group is a group that is a media intelligence company. They have 55 offices across 6 continents. They do global media intelligence, and what they tell us is that the whales in the Salish Sea are globally interesting. Some of this is just a few months last year, but you're looking at that spike there. It's about 300 million people in October, just over a few week period in October, and the other bites are just, you know, other related stories. This is a heat map. We're big in China, we're big in India, we're obviously big in North America, and the Chinese really are interested in our whales. They're picking up a lot of stories, and that's just that heat is getting redder and redder on that heat map. Scooching through here, that shows that basically over a 5-month period that we reached 2,071,000,000 people globally in every developed country in the world with the media that we were advancing. And those are largely original articles that they're picking up or rewriting or retranslating. Very, very big. I'm going to push through here a little bit. This is sort of recently, but I do want to show you this August bump right there. You see that bump right there? That is when we put out a couple stories about humpbacks. So that's largely humpback media right there. And over the course of just a few days, we reached about 250 million people worldwide on just some humpback stories coming out of the Salish Sea here. So humpbacks are big, big. And, you know, and you see a snippet there of the baby orcas. We had 10 births over the course of 13 months with the southern resident community, and each time we put a story out announcing the birth with the Center for Whale Research. People got invested around the planet. People are really watching this population of southern resident killer whales and rooting for the babies. All right, pushing through here a little bit. Reach there, yeah. So basically over the course of the last year, we're about 2.5 billion people around the planet have seen the media that we've done. And I think it's very positive. The stuff that we've tried to put out are all about not just what we're seeing, but how can we protect what we're seeing. Audacity of hope. This is where sometimes I get into trouble because we have a population now, it's down to 81, 80 if you count J54. Being missing. I don't think J54 is officially missing yet, but just not doing very well. But we're down to 80 whales on this population. I was one of the petitioners on the ESA case and actually a litigant in the U.S. District Court that got them listed. And we were at, I think when we first put it up, we were 78 animals, and now we're at 80. So we're not getting much done here on this recovery. But the one thing I know in doing the media I've done and speaking for this association is that if you don't give somebody a sliver of hope, it's really hard to get that global investment, those people outside of this region supporting recovery efforts and just, you know, the broad support that you need to recover this population. So one of the things that we did back in October was we kind of just collected all the photos and all the most recent reports that we had off the water of the 8 calves that were born between December 30th of 2014 and January of 2016. So over a 13-month period, we had 10 births. 8 of those whales survived. And depending, again, if you count J54, 8 out of 10 over 13 that we know of, and a population as prey depleted as they are, I'd say that's a little something to hang your hat on. And so that's what I call the audacity of hope. Again, though, we called it the class of 2015. But again, it's just, but the orcas are still in trouble. They still need fish. But here's something we can hang our hat on. So pushing through here. Another thing of hope, the audacity of hope. And I know this is a tagging story, but No Fisheries came out, announced necropsy done on L95, and this is the animal that was found in March, I think March or April, and tagged that had the petals still in it. And the necropsy came out. They did a science panel also on this, and they announced the release. The interesting thing is that before they announced to the media what the results were, they asked Ken Balcom, Deborah Giles, myself, and Jenny Atkinson from the Whale Museum and the Center for Biological Diversity, Miyoko from CBD, to get on a call where they, 45 minutes before their announcement, to just talk about it. And what came out of that meeting is a real sense on No Fisheries that at least I felt like they're finally getting it. They're finally understanding. They're finally going to start moving on this thing. So out of something bad, I have hope that we can get something good out of this. And so I can address that a little bit in Q&A. So I'm almost done here. But here's the other thing I'm really excited about. Being from Washington State, and I had some great discussions with some Archie from First Nations here, and we have down on the U.S. side the Bolt decision. That was 1974 and upheld in Supreme Court in 1979 by my former father-in-law. But it's the tribes in Washington State, the 20 treaty tribes of Washington State are co-managers of the fisheries. They're another government. There's oversight, but also equal authority on many issues involving fisheries. And the Obama administration, again, saw this as an opportunity to leverage these treaty rights to start recovering salmon habitat and get some fish in the water. And I think there's going to be probably $500 million U.S. going into this effort. We're going to start seeing things happening in the Puget Sound that will help southern resident orcas. That's it, and just PWWA going forward, and I'm going to kick it to Tasley one of these days. But following C, I think that we are set up right now, and as I depart Pacific Whale Watch Association, I think we do have a following C. I think there is some positive things that the association can build on. I don't think they're replacing, there's not going to be another executive director, but there's a lot of really progressive people within the association. I urge all of you to reach out and talk to them. Now, I want to kick it to Tasley Shaw.
Thank you, Michael. Just like really quickly, first of all, my heart does go out to the Vancouver Aquarium family, period. And second of all, Casey was supposed to be here giving you guys a really rich, interesting presentation about all the really specific things that we've been seeing out on the water this summer, complete with photographs and all the rest of it. And I was here for kind of the fluff part of it. And as you can see, Casey's not here, so you're just getting fluff. So sorry. In addition to working as a naturalist for the past 7 years, I'm in my final year of study at Emily Carr University, and I'm doing a degree in illustration. And basically all of my images are of marine life, and I like to take the observations that I have out on the water and bring it back into the studio. So I thought I would just really quickly show you some of the images I created over this season and give you a bit of context. So this is Group A of JPod, and I think that makes sense to point out that we don't very often see JPod all together as a group. At least in the last few years, they've been sort of traveling as these two groups, which we kind of arbitrarily call Group A and Group B, and I think that's fairly significant and an indication of these changing social dynamics that we're seeing in the population as a whole. And understandably, there's been a lot of attention over the upsetting disappearance of J28 and very likely her calf J54, and I think that's kind of overshadowed a little bit the disappearance of a really great matriarch J14. She was last seen in early August and hasn't been seen since, and she was a 3-time mom and actually 4-time mom and a grandmother, and she's kind of one of those woo-woo whale magic kind of individuals for me, and I felt compelled to do this sort of portrait of her, and I'm really interested in doing life-size work, which you'll see later on. And this has since been printed out life-size, so you can have I guess a staring contest with J14. So we're all super excited, and I've been blown away by what's been going on with the humpbacks out there, and I feel really, really fortunate that I've sort of been here to see the change in the humpback abundance in the Sailor Sea. And these two individuals that you see are, one of them, the one closest to the camera, is a whale that's pretty well known to us out on the water. That would be the very creatively named split fin, not to be confused with split fluke, by the way. It's an individual that we've seen over several years, and the individual at the back is one that's kind of a new kid on the block, and that's becoming a trend that we're getting new individuals every year. The whale in the back, by the way, known as two-spot, and I spend a lot of very late nights, 4 a.m. kind of thing, looking at everybody's pictures on Facebook of the humpbacks that they're seeing out there. And trying to match them up, and if they've been seen elsewhere on the coast. And two-spot has been seen quite a bit in Clayoquot Sound, which I thought was interesting. And for those keeping track here, this is Split Fluke, Big Mama, and Heather, and they represent 3 generations of humpbacks, which I think is kind of a great. They're icons in our area, and I think a really great way to represent how the population as a whole in the Northeast Pacific is making a comeback.
And finally, this is my senior project this year at school. I've created a life-size, basically a digital mural of J-Pod that I've very obsessively, meticulously represented each whale as closely as I can to how it looks in real life. So not just dorsal fins and saddle patches, but eye patches as well, and any particular rake marks or anything like that. And sort of the whole impetus for this was to have it shown in a public space, hopefully in the future. So stay tuned for that. And it really all started because my initial fascination with killer whales started when I was in 3rd grade, and my teacher told me that their dorsal fins can be 6 feet tall, which is as tall as the principal of the school. And that like blew my mind as a little kid. So I would love to somehow represent a life-size killer whale for the public without having to add any additional impact to them. And I think as we're all filing out, I know like there's pizza and beers waiting. So I'm not gonna show the piece for you right now, but we'll put it up on the screen and play it as you guys are filing out. Yeah, so thank you.
I just got to say really quickly, part of the reason why I wanted Tasley here is because I'm a big fan of her art, and it's amazing how her experience on the water, and I know she's getting old, informs how she does things, and it's just, that's beautiful stuff. So
Thanks, Michael.
Yeah, no, it's beautiful
Thank you.
work. Do we have any questions? You touch on so many things, Michael.
Yeah.
We got one online question.
Yes.
Need the mic?
All right, so we have one question from Celia, and she's been patiently waiting for the answer to this one. She just wants to know whether the association educates members about the impact of the issue of exhaust from gasoline engines sitting on the surface of the ocean, impacting and its impact on the whales' breathing.
Interesting. I've got to say that that's sort of out of my wheelhouse a bit, but I will say that I found the association to be very progressive on acoustic inputs over the years, and watched the fleet modify, and some of the, we talk about the profits that are going into the association. Many of those profits aren't just buying new fancy boats, they're going into modifying the boats they have and trying to reduce the soundscape that they're generating. And back in the day, when I first started with them 5 or 6 years ago, you had a lot of people that basically just were deniers, just didn't feel like what they were doing was putting anything or having any impact on the whales. And it's a whole new ballgame now. I think it's really a very progressive group out there among, you know, you got 37 alpha males largely and spouses. So it's tough to get one collective thought, but enough progressive people and enough modifications. But I can't answer anything on engines and diesel exhaust.
Anybody else?
Questions? Take that one, Tesla.
Hi, I was just wondering if you could speak to how the whale watch operators, what they're, if they're being receptive to the idea of not just converging in one area on whales, because I live on Saturna Island out near East Point, and there was a couple times this summer where it was really disconcerting to see the amount of whale watch operators on, you know, like 12 boats on 4 whales, and just pinning them up against the shoreline, and it was really hard to watch.
Yeah, it's hard for anybody to watch, and I think that that's kind of the undercurrent here. It's not pleasant for the crews, it's not pleasant for the passengers, and so that's when I talked about spreading out the fleet. Again, it's also economically, it doesn't make sense for 21 different ports to all come. We got people at Cowichan Bay, we got people coming from long distances, traveling over and burning a lot of fuel to get to an area. One of the big problems with that is that when you get these boats, these whale watch boats that are coming long distances, they just have a short amount of time to see a whale, then boom, they got to get out of there. And so one of the problems that we were having, and we noticed several years ago, and I think we've addressed pretty well, was our approaches and departures. You know, people zipping into a scene, right? And then once they're done, oh my God, I'm over time, I got to get them back, and then zipping out. Well, all that stuff is really the primary factor on D-tags. what we learned from DTAGs, the analysis that NOAA Fisheries is finally getting around to doing on DTAGs. But one of the things that they said, their original analysis, was that the number one criteria, the number one impact thing for putting sound in the water is speed, vessel speed. Well, we addressed that several years ago, and we still got, a couple people that need to kind of get the memo and reread it. But I think we've really decreased our vessel speed, and I'd like to see it going forward. Talk about the following sea. I'd like to see going forward there, speed limits, some real focus on speed. But spreading that fleet out with humpbacks, fin whales, dolphins, gray whales, it's really helped the industry economically, and it's helped the whales. And it's helped southern residents, I think.
Michael, is there any discussion in the association about no entry zones?
Yeah. Oh, absolutely. Yes. Yeah. Well, within hours, I think, after I announced I was stepping down from the Whale Watch Association, the no-go zone off the west side of San Juan Island sort of resurfaced. And this is a closure zone primarily for commercial whale watch vessels off the entire expanse, and I think out to a mile in certain areas. Well, there's already off the west side of San Juan Island, which is kind of Orca Central in the middle of summer for southern residents especially. But yeah, there's been a proposal. Interestingly, I met with them a couple weeks ago at their request. They came to my office in Seattle and wanted to talk about it. I think there's a lot of people within the association who understand the importance of MPAs. I think that the problem that the association had with this was, number one, calling it a whale protection zone, not an MPA. We're protecting a lot more than whales if we're closing areas off. But there's some serious discussion now, and I'm supportive of certain areas, looking at certain areas, and working with people like in this room here, to come up with the best way to do set-asides that actually will work for the whales and won't just be optics. That's a concern a lot of the whale watch operators have now, is that a closure zone off the west side is just optics for some people. It's just getting whale watch boats out of the view of certain people. And, and some of those proposals have been advanced by wealthy west side San Juan Island landowners who don't want to look out their kitchen window and see a whale watch boat. But what we learned from DTAG and what we're learning, it's not optics, it's sounds, what's going on. It sometimes doesn't matter as much how far away a boat is. What matters is if it's spinning props, and what kind of sound, what kind of approaches and departures the boats are doing. What kind of sounds is it putting in? I answered your question.
Any other questions? Yeah, I think I'm now going to thank you both. Well, and we are going to play.
Yeah.
Piece,
so
I may just have you stand. So good at standing.
Okay. Yeah. Got to get used to it when you're with Michael.
Yeah. We did just a couple of announcements. One is next year's symposium will be, whoops, that's my phone ringing. That one.
Sorry, I gotta take this.
No. Sorry. I'm back with you. Our next year's symposium will be Saturday, November 25th. So if you want to mark your calendars now, that'd be great. We also have one notice here from Joe Bauer. And I'm going to just pass you the mic. Joe, I'm not sure how many of you know Joe. Somebody who comes regularly to our seminar here at the university, former fisherman, and he was a big part of the original, the first capture of a killer whale being brought into the Vancouver Aquarium. But Joe's here to talk to you about herring.
Well, as you're all aware of, it's fall. And fall is the best time for herring as far as their taste and the oils, and including omega-3 that they contain. So the fishermen get together this time of the year and use their vessels, their gear, pay for their own fuel and everything else to go out and catch herring to help kids with cancer. So the herring will be on sale December the 3rd, which is next Saturday, and from 0900 hours to 1600 hours, or 9 o'clock to a.m. to 4 p.m. for those of you that don't go on the 24-hour clock. And it's up at Trites Road in Richmond. So if you're coming out to Steveston, and you're coming out number 2 road, you just keep on going south on number 2 road until you get to Moncton. Then you turn right on Moncton, which is the only way you can turn. You end up in a ditch in the other direction. And you go down to Trites Road and then head south right up to the wharf, up to the Harbor Authority. And if you're coming in one road, then you go along number 1 road until you hit Steeston itself, village, then make a left turn and head up Moncton Street until you hit Trites. Same thing again. The price is $15 for a 20-pound bag of really delicious and edible herring. They're the best this time of the year. They're really delicious. The money, every penny of it, or every dollar, goes to help kids with cancer. It's the fishermen contribute their time and their vessels and their fuel and everything else to that as their part of the effort. And then you have a chance to contribute a little bit of money for the kids with cancer. Okay, I'm going to cut it short.
And Joe tells me you don't have to be Norwegian to buy herring and pickle it. Really good. And I would encourage, for those of you here who have not met Joe, to take a chance and ask him about experience with Moby Doll. And I first heard it from him, and it's quite a remarkable story. So I'm now just going to, before we run the final credit, just thank a few people. Last night, I guess we were here until about 10 o'clock. Marcus was here, and Hunter at the back, as we were running around trying to break through firewalls and get everything wired to work. And Pamela's here with us as we were looking for everything. And miraculously, we made it work again. This morning, though, it took Rob to come in to really break the firewall to get us through. And, and the one thing about having Rob come in is that he just calms us all down, because we know he's going to make it work. So I'm particularly grateful for the three of them for coming in and running the high-tech part of this that in the end makes it look simple, but I was sweating it this morning. Were you? Okay. Very calm, very calm. Also want to thank David Rosen and Kate Keogh for pulling together to get food. Kate has done this for 24 years from the very beginning. Although as it's gotten bigger, we're now sharing some of that with others. In terms of getting it set up and registration, I've got a lot of the members here of the Marine Mammal Research Unit: Selena, Hassan, Brianna, Cody, Mariana, Aaron, Ria, and Beth. And they've all pulled it together for us. Thank you very much. I've got to really thank Pamela, who, you would not believe how many hours she puts in to trying to make sure she got everybody's name on the list, get the name tags made, and even though I got blamed for making them crooked, we still pull it off very well. I want to thank the people who came forward to speak. We really do appreciate people sharing their knowledge and telling others what you're doing, what you're learning. And for sponsorship, again, we're getting more and more people, so I may be asking for a little bit more next year. Just a heads up to the Pacific Whale Watch Association and Pacific Wildlife Foundation. Thank you very much, and also all the members of the Marine Mammal Research Unit. With that, we're going to set the credits running here. I shouldn't say credits. I'm going to let you just set it up here. But just thank you all very much for coming. I don't know what we came to in terms of final numbers of people that it came in at, but it got up 2,110. Awesome. Yeah, so all unique individuals. So who would have thought? But anyway, thank you all. Please stick around and chat a little bit. And Lee, I'm going to let you just say a little bit as we play that. Maybe we'll just dim the lights too so we can see it properly.
Okay. Oh, thank you. Well, there is a sound component. It's the calls of J-Pod that a couple of friends of mine donated to the project. Yeah, so I call this digital whale watching.
Thank you all very much.
Perfect timing.
What do you think we try to do for your art?
That would be amazing.